closer adjacent spatially to agricultural fields, and that the strongest selection was among those genes and alleles that were specific to herbicide resistance in plants. So here we can see the kind of human fingerprint of how agricultural applications and the application of herbicide especially is shaping native plant diversity. And finally, this is an instructive and sort of sobering example of the way in which humans are apparently shaping the loss of genetic diversity in natural plant populations. This was a demographic modeling study using genome sequence data from herbarium specimens involving the model species Arabidopsis. And here, these researchers identified the loss in about 80% of the standing natural genetic diversity of this plant species, especially in parts of Europe, and projecting that forward under future losses are even more dramatic in terms of the loss of genetic diversity in the Anthropocene. Similarly, we can utilize these collections for investigations of microbial diversity. This is one of the kind of newer, more exciting applications, but here to study pollen transfer, to study internal and external symbionts, to study interactions with insects. But the idea is to use antique herbarium specimens to study interactions between organisms, especially involving microbes. And one of the first of these applications, indeed, the first to decode the genome of a plant pathogen from a herbarium specimen involved the fungal potato blight that led to the famous Irish potato famine. And here, these researchers sequence not only the plant, but the pathogen to identify that the pathogenic strain arose in Mexico, far from its Andean origin, and moved from Mexico into the ports of Western Europe well before it became problematic and a source of famine that would decimate parts of Western Europe, especially in Ireland. So novel application of herbaria to study not the plant per se, but some of the pathogens associated with these plants of human use. Similarly, to study symbiotic interactions, cryptic symbiotic interactions, these are ones of a yam species widely distributed across Africa and Madagascar that has associations, obligate associations with bacteria. And here by sequencing the plants, the bycatch of this also highlighted a complex series of interactions involving vertical transmission as well as horizontal transmission of these symbionts for this plant species across its distribution. So now I'm going to transition to the first two parts of my talk to moving to an orientation of the broader applications of herbaria for societal issues. And I'm going to start with this study of ours that looked at the colonial legacy of herbaria. And here we utilized 5 million herbarium collections to characterize the point of collection and the movement of these collections around the world from their origination point. And what you see here is very much the historical impact of colonial era collecting and the development of herbaria, especially in the global north. And so the red indicates collections that have moved from regions predominantly from the global south to parts of Western Europe, and those in blue from the global south to areas of the US. Similarly, there's movements into parts of Asia. But mainly what you see is that the majority of herbarium collections are in the global north, and that's odd because most biodiversity is in the global south. So there's almost an inverted biodiversity gradient in terms of where plant biodiversity lives and is found natively versus where it's housed and studied in herbaria. And this colonial legacy has continued on beyond the formal ending of colonial periods. Obviously, um, these days there are a lot more checkpoints in place, and collaboration is the key in terms of moving forward. But nevertheless, that legacy is always present and part of these collections. The other element of Herbaria for Society is trying to bring these collections, digitized or otherwise, into classes for higher ed university students, as well as STEM students in grades 9, 10, 11, and 12 in particular. And this is a class that I began during the pandemic, highlighting the novel relevance of natural history collections to phenomena and questions during the Anthropocene. I built a series of modules utilizing the physical collections, and it was really born out of the motivation that most students love collections and museums of various sorts, but are unaware of the research agendas and the research parts of these collections. So this was all about exposing students at Harvard to these collections. We've now broadened these curricular modules to educating high school biologists in grades 9 through 12 to bring these kinds of resources into their classrooms, especially through the use and application of digitized collections. And this has been enormously successful. We've now done these workshops across the United States and it gotten incredible feedback along these lines. Similarly, I'm very excited about the intersection and application of art and science, and especially on the use of art for as an untapped resource of biodiversity. This is exhibit going up featuring Rachel Rice's collections at the Museum of Fine Arts, Boston. It's currently showing at the Toledo Museum of Art in the United States and previously was exhibiting at the Alte Pinakothek in Munich. But basically, this highlights a retrospective of Rachel Rice's work, this botanical still life artist. And what we did was to inventory in a very detailed, sort of floristic way, the paintings that she depicted. And what we are able to sort of characterize is, the colonial history of botanical exploration, especially as led by the Dutch. And here you can see that the distribution of plants that made their way from their native environments across, especially regions of the global south, to her canvas in Amsterdam were facilitated by the activities and the actions of the Dutch colonial empire. And you can see that the diversity that Raj painted throughout her career exhibited a sort of hump-shaped distribution where the highest diversity paintings she created were in the mid-period of her career. Indeed, the highest diversity painting shown here may have been related to the passing of her father and may have been an homage to him, who was very much a famous and well-respected botanist and interacted with various scholars across Western Europe, describing biodiversity as it came into Europe. So I hope with these various kind of highlights, I've convinced you that the Global Metaherbarium is truly, designed to create linkages both to the digitized specimens as well as the physical specimens to expand science in a way that we never imagined impossible. And as we move forward in the future, I'm constantly struck by the fact that we're continuing to innovate novel means and methods of utilizing these collections and in ways that we never imagined, and that there's responsibilities that we have to utilizing these collections in these novel ways. I've written on that as well, but won't talk about that much here. But nevertheless, it's an exciting time for herbarium science and herbarium practice. So now I want to finish the last part of my talk, which is germane to Nigerian herbaria and Nigerian biodiversity scientists. And this is part of a phenomenon that we call the silent herbaria. And this was a project led initially by Daniel Gigela with support from Ryan Schmidt. Daniel visited my lab, and this was enormously productive and a rewarding exercise. But silent collections, and this kind of evolved in conversation with Daniel, are really ones that are relatively unknown, they're underutilized, and they're underappreciated by the global botanical community. And it's important to recognize that silent herbaria exist around the world, but in these conversations, it became clear that places like Africa might be especially high in silent herbaria. And so we mobilized a team with Daniel's good support of about 24 biodiversity scientists in Nigeria that are herbarium directors, curators, staff that steward and support these collections to begin an exercise to explore this. And the foundation of this work was really based on the premise that among those collections registered in Index Herbariorum, this is the central repository for recognizing institutionally at a global level herbaria, that new African herbaria were modestly reported since that period of time. So within approximately the last 10 years. And so you can see that over this period in the global south, not quite half of the total herbaria newly registered were from Asia, South America, Central America, and the Caribbean, and Africa. And lowest on this list was Africa, which registered just under 50 new herbaria, or about 6% total of all new registrations. Now, it may be the case that this accurately reflects new herbaria, but we wanted to explore this. And in particular, we were interested in the question of whether silent herbaria were especially prevalent in Africa. And what we found through these surveys and within our collaboration group is that indeed herbaria were woefully unrecognized in Nigeria in international registries, in particular in Index Herbariorum, which tracks herbaria. And so here you see a map of Nigeria, and the open circles are those herbaria recognized internally, but which are not recognized in Index Herbariorum. So at the most fundamental level, these are silent herbaria, and this includes about 80% of all the herbaria across Nigeria. And they are then effectively cut off from international collaborations that are facilitated through this important registry, and indeed through restrictions to some funding avenues and venues. And this is especially surprising because despite the underfunding and some of the concerns regarding the infrastructure that support and sustain these collections, they've grown enormously over the last 25 years. So here we see reporting from our survey recipients. The red line tracks the growth from those scholars in Nigeria. And the blue line captures the much more modest growth as reported through the international registries in Index Herbariorum. And importantly, what we can see is that the internal growth of these collections greatly exceeds the growth of herbaria in all across all of the planet during this period of time. In other words, the growth in Nigeria herbaria has been truly exceptional, and we documented all sorts of important applications of these herbaria for research and especially for teaching. And importantly, we conducted a survey of the biodiversity modeling that can be done with these in-country collections versus those Nigerian collections that reside out of the country. And I should add here that among all the 500,000 or so Nigerian plant collections, only about 10% of these are digitized, so a very modest amount. And even with this modest amount of digitized Nigerian collections, you see fundamental differences in the application of in-country Nigerian collections versus those out of country. So here we see enormous temporal expansions when you consider in-country Nigerian collections. So here, between years, you see in particular that recent years over the last 20 or so years are much better represented by Nigerian collections. This is obviously very important for climate change investigations and how plants have changed over recent decades. And we also see this phenomenon when looking at collections activity within a year, and indeed some months are not even represented when you look at collections outside of Nigeria. We think this is a result of parachute science and academic semesters outside of Nigeria in and around parts of the global north. So enormous temporal expansions facilitated by these in-country collections. Similarly, when we look at various species and conduct species distribution modeling, we find out that those ranges are much more realistic when contrasting the in-country results of the models shown in figure G here versus the out-country models. So the idea here is that these collections, which remain silent, are really essential for biodiversity modeling, and it's important that we sustain and steward those collections more effectively. And one of the first and best initial ways we do that is to get those registered. Registration is not hard, it's free, it's really a simple email to recognize that these collections exist and the point persons that are responsible for stewarding those collections. So many thanks for your attention. This has been an enormous pleasure. Thanks for accommodating the hybrid model here for the presentation. I very much look forward to the question and answer session that follows, and I want to highlight these papers for future exploration. The Herbarium of the Future, which is a paper that I published a few years ago, a recent paper on the guidelines for the effective and ethical sampling of herbaria, and the recent paper led by Daniel Gigela on silent herbaria and with a test case for Nigeria. So thank you so much, and I look forward to the questions that follows.
I believe the keynote speaker deserves another resounding round of applause. Thank you very much. It will interest you to know that some of the participants here today are not just from Botanical Society of Nigeria, from the field of study of plants-related study. We also have renowned professors from different field of studies. For example, our acting vice chancellor is a renowned professor of law. A round of applause for that. She's a renowned professor of law. We have professors from different field of studies, from arts, engineering, other science-related courses. And I want to say that as much as I have learned a lot from this presentation, I believe that we all have learned something from this. Have we? One major thing I learned, or that I was made to understand, is the concept of herbarium. Before today, I didn't even know what herbarium meant. I didn't even know that we had something called silent herbaria. And with the data that our presenter gave to us, we saw that there is a lot of work that needs to be done, especially in Nigeria. As a society, Botanical Society of Nigeria, we can see that we have a lot to do. The presenter made mention of we having over 500 plants collections, with just 10% of these being digitized. Like our acting vice chancellor mentioned, that we hope that this conference will bring collaborations, will birth new ideas for us to collaborate and bring ideas that will help the plant and environment study in Nigeria, because plant is a very important aspect of human existence. So we hope that this conference will bring new ideas, we bring new themes, everything, collaboration that will help the society, Botanical Society of Nigeria, and plant and environment as a whole. Thank you once again. A round of applause for our presenter. Thank you very much, sir. I'd like to crave our indulgence. The acting Vice Chancellor has another engagement and would be taking a leave now. We'll just kindly take a group photograph with members of the I table before she takes a leave. A group photograph. Yeah, members of the I table and the council members will join. Members of the I table first. While this is going on, I'd like to acknowledge the presence of some dignitaries here today. Firstly, I'd like to acknowledge the presence of the Dean of the School of Postgraduate Studies of Benson Idahosa University, Professor William Odo. You're welcome, sir. A round of applause for the Dean. It will interest you to know that one of the venues for our breakout sessions will be held at the School of Postgraduate Studies. We also have
council members, please join.
The Director of Academic Planning, past Dean of the Faculty of Agriculture and Agricultural Sciences, Benson Idahosa University, currently the Director of Academic Planning of the University, Professor Ellen Ajayi. You're welcome, ma'am. I'd like to reintroduce the representative of the Acting University Librarian, Dr. Mrs. Mary Idahosa. You're welcome, ma'am. We have with us the Managing Director of Ogba Zoo and Nature Park, Mr. Andy Ehanire. You are welcome, sir. Welcome, sir. Thank you very much for being here. Just for our information, questions will come after the plenary speaker.
Can Professor Odu, ably represented by Dr. Ngozi and the HOD for Biological Sciences, please quickly join. Please do not leave, ma. Sorry, ma. Okay.
Okay, can we rise as the Vice Chancellor takes a leave? And give the Vice Chancellor and the entourage a round of applause for being here, for gracing us with our presence.
Thank you.
Thank you very much, ma'am. Thank you, sir. Thank you.
I also like to recognize one of our distinguished professors, the Director of Research and International Programs, Professor Falodu. Please celebrate him. Thank you, sir, for being here. Thank you so much. Thank you. Thank you. We would love to have a group photograph with every participant here and delegates outside. Yes, so please, I'd like you to just quickly, yes, Mr. Chedrak, your attention is needed outside. Can we just leave our bags and then just go outside and then we'll come back quickly? Yes, can we do this now? Yes, while they're still outside. Thank you so much. Please, Mr. Shadrach, I'll need you to organize this. The whole participants are coming outside for a quick photograph. Thank you. Thank you. Please, can we do this quickly and then come back so that we can have the plenary speaker take his presentation. Thank you so much.
One of the things that we have desired about BIU is to make BIU unique, innovative, and do something that is not done by other universities. The type of graduate we need in Nigeria is not somebody that will just graduate from the classroom, have a BS, and it ends there. No. We have believed that Benson Idahosa University to be another university where other ingredients of success in life can be put into students.
What differentiates a good editor from an editor is how creative you can piece those footage together and make sense out of it. The color is in charge of evoking emotions using color on screen. Some elements of film editing include cuts. You know what cuts are, like the footage itself. You have to cut it somewhere. You have to join it to another footage that you likely have cut somewhere. Pacing. Pacing is the overall feel of the film. Continuity is important. It should follow the way your normal life would
And I'm so happy to be here. This is the day 3, and I've learned so much from the first day to now. And presently, we have learned so much about storytelling, cinematography, and a lot of other variables in the entertainment industry.
From a level of knowing nothing, I know almost enough to film a good movie.
I've learned a lot of things from storytelling, script writing, and video editing. It has been so wonderful.
One of the things that we have desired about BIU.
I'm Dr. Femi Idahosa, President of Benson Idahosa University. Now at Benson Idahosa University, that's BIU, our message is clear, empowering you for global success, academically, socially, and spiritually. We are committed to raising a new generation of leaders. Once again, that's you. Young people equipped not just to succeed, but to transform the entire world. Our graduates are living proof, making impact across industries and continents. They inspire us every day. Now to our current and incoming students, you are the agents of change the world needs. We need your voice, your vision, and your values. And the time to take ownership of the future is now. Behind every student's success is our dedicated staff of mentors and educators who believe in each student's potential and work tirelessly to bring it out. Their impact is real. We are more than just a university. We are a launch pad for greatness. And with each passing year, I grow more confident that we are doing something extraordinary. Because at BIU, everything we do is focused on one thing: empowering you for global success.
I got to find out about Benson Idahosa University through my parents, and when I got here, at first it wasn't easy because I had to adjust to a new style of doing things, a new environment, and meeting new people, and having to go to chapel every Friday. However, since that time till now, I've experienced all-round growth. I've grown in my spiritual life, my academics, my social life, and in my interactions with people. Since I got into BIU, I've been involved in different areas of the school, especially within my faculty. I was the Director of Works, the Assistant Secretary General, and now I'm the Secretary General of the Faculty of Law, Benson Idahosa University. And if I was to choose another university to go to, I would choose Benson Idahosa University 100 times over.
Hi, my name is Agari Ogenemaru Daniel. I'm a 200 level law student. I got to know about Benson Idahosa University from a friend of mine, and he told me about it, and I said, fine, let's try it. And since then, my experience has been very interesting. I got to join the choir, and I also got to be part of the student leadership body. And it has actually helped me to discover my potentials and more things I can do as a singer. If I was to make a choice to another university, I would definitely and gladly choose Benson Idahosa University.
Hello, my name is Joshua Tamunateli, and I'm a proud student here at Benson Idahosa University. I had my reservations about enrolling into any private institution, due to how one can feel restricted. And to be honest, I was wrong. The decision to come here has turned out to be an incredible journey. Benson Idahosa University has helped me grow beyond just academics. Here, I found my voice, built strong friendships, and learned under lecturers who consistently challenge us to think critically and become innovative. I came into Benson Idahosa University knowing little or nothing about life, but now I am a student leader. BIU is definitely shaping me for something bigger than just having a degree in my field of study. It is setting me up for global success.
Hello, my name is Enakiri Desiree, and I am a proud student at Benson Idahosa University. When it came to choosing a university, I wasn't just looking for a degree. I wanted a place that would sharpen my potentials and align with my values. Benson Idahosa University stood out as that place. Here, excellence isn't just expected, it is nurtured. Our lecturers are mentors who challenge us to think deeply, lead boldly, and grow consistently. What sets Benson Idahosa University apart is its foundation in faith. It's an environment that builds both competence and character. I'm not just gaining knowledge today, but I am being prepared to make impact, to lead, and to innovate. Benson Idahosa University is truly preparing me for global success.
God sends abundant life to you in the name of Jesus. From the word we heard today, we will give you abundant healing in Jesus' name. From the word we heard today, we will give you an abundance of His grace in the name of Jesus. I prophesy to you in the name of Jesus. Everything God has spoken, we will see come to pass in the name of Jesus. Grace goes before you this week. His power will speak for you this week in the name of Jesus. Abundant life shall pursue you. Abundant life shall find you. Abundant life shall be your portion in the name of God the Father, God the Son, and God the Holy Ghost. We pray. Amen.
The things that we have desired about BIU is to make BIU unique, innovative, and do something that is not done by other universities. The type of graduate we need in Nigeria is not something.
Thank you and welcome back. I can see that we're all, our mouths are busy, yes, for some of us. I think we had to decide to take the break at this time because it was such a beautiful sight to see every one of us, socializing, connecting, networking. I heard one participant say, oh, I haven't seen this colleague in since 2013. Another one said, oh, this is my classmate. I saw him last in 2009. So it's such a beautiful experience. I saw some beautiful ladies, participants over there taking some selfies, So I can see that we're actually very happy to connect again with one another. And I think that's the beauty of such a conference like this, bringing people from all over the state. So we're glad and we're happy that you have the privilege to be able to enjoy that moment of fellowship with one another. But however, we're going to be going forward because now we're going to be taking the question session because our keynote presenter is online with us now, ready to take our questions, Professor Charles Davis. But just before that, I'd like to welcome the president. Yes, the president is already here. Please mention some of our dignitaries to join us in the high table. Yes. Okay, Professor Falodun, sir, we'd like you to join us on the high table. Professor Iboka, please kindly join us on the high table. Okay, and this. Okay. Can we have these dignitaries join us on the high table, those that I mentioned? Professor Falodun, Professor Esther, yes. Adelawa, yes. Adelawa, can she join us on the high table if she's here? And Professor Emmanuel Ibokan, yes. Please join us on the high table. Thank you so much. A round of applause for them. I know your hands are busy, but please try to, yes, celebrate this. Oh, thank you, sir. I quite appreciate that. Thank you. Thank you so much. Thank you so much. Thank you. Professor Esther, so highly welcome, ma. Thank you. Thank you. Professor Faladu, thank you. We want you at the high table, sir. The Director of Research and International Programs. Thank you. So please, at this moment, I'd like you to take note of your questions, whatever questions while the presenter, keynote presenter was giving his presentation. You had specific questions that you love to ask. He's online with us now, and we'd love to take those questions. Also, not just the physical participant, the virtual participant. Professor Davis, we're glad to have you with us online. Thank you so much for joining. We quite enjoyed your pre-recorded presentation earlier on. Thank you so much. It was quite educative, and we learned a whole lot. And we're going to be asking some questions, and we know that you will give us answers to our questions. So please, I'd like you to take notes. If you want to write out your questions, no problem. If you want to speak, he's all ears, and we'll progress. President, sir, I'd like you to also take charge of this session, okay, while we'll step aside. Yes, someone to share the plenary because that's all right. No, the plenary session. Okay. Okay. So, yes. Yeah, okay. So we'll just take the question and answer. Much later, we'll have the plenary session where Professor Ibokan will handle that for us. So please, we'd like to just, if you have a question, with regards to the keynote presentation that was done by Professor Davis, Charles Davis, I'd like your hands to be up so we can identify you, okay? We can identify you. Please, yes. Yes, I see 1, 2 hands. Oh, 3, okay. Please, I'd like your hands to be up so that we just take them quickly. Because we can't take so much, we just take a few so that we can move on. Yes, we'll start from the high table. Professor Iboka, thank you. Yes.
Thank you, Dr. Davis, for a very detailed, comprehensive, and inspiring lecture. I want to thank you for taking in my friend Ziegler for the work, the beautiful work you have done on silent Iberia in Nigeria. And such opportunities are actually important, especially for some of us that probably find ourselves in this silent mode in the global biodiversity firmament, if you like. But I want to say that as many as such opportunities present themselves, people like Ziegler and other young Nigerians who have a flair for taxonomy should be given those type of opportunities to expose themselves to the basic technicalities that are involved in standardizing our taxonomic work in Nigeria. It is important for you to know that taxonomy is not one of the best subjects that a typical botany student in Nigeria aspires to undertake, because it is they find it very daunting task to get involved with the nitty-gritty in plant identification because of the technicalities involved and the lack of resource materials by way of books, illustrated guidelines, and the like. Yeah. Most of the books we use for taxonomic studies in Nigeria are actually sourced from authors like yourself in the temperate regions. But except for those who have come here during the colonial times that have found time to write our local materials bequeath on us those type of works. We have very few local inputs. But Ziegler also has produced a very beautiful illustrative guide for savanna plants. Such books and such technical know-how are very important for us to move away from this silent mode into a more visible mode. So I want to congratulate you for this work, very notable work, and hopefully it will ginger some of us who are not scared of taxonomy, biodiversity, or conservation to take it to the next level. So thank you very much for your efforts. Thank you.
Well, Professor, and good afternoon, everyone. It's a pleasure to be here. I want to thank you for your comments and considerations. It was just such an enormous pleasure to work with Daniel over the period of 6 months when he visited our lab, and it was indeed very fortunate that Harvard University has provisions and support through their Center for African Studies program to help facilitate his visit. Obviously, from the tone and the theme of my talk, it's clear that herbaria are a central pillar for the research that I do, whether it's taxonomy or molecular evolution or evolutionary ecology. And I think that for 30 years, I've focused on research that centers herbaria and working with colleagues to share the joy, the excitement, and the beauty of these collections. And my hope, of course, is that through digitization, we're better able to democratize knowledge and information. And it was really very organic through conversations with Daniel that it became clear that this was very much an area of biodiversity science and herbarium practice to explore, and through more than 24 Nigerian collaborators, scientists, curators, staff that work at these collections, that they're centrally important to understanding global biodiversity. And working towards sort of drawing out that story and that narrative was very important, not just for Nigeria, but for thinking more broadly about these sorts of collections across Africa, across the global South, and indeed across many of our collections in the global North. I hope it was clear from the talk and from conversations that you may have had internally that silent herbaria exist everywhere around the world. In my home collection at Harvard, only about half of our collection is digitized. We're working towards completing the entirety of the collection and making it accessible online to the world. But at present, half of those collections are silent. So these collections exist everywhere, and making them accessible is really one central element to democratizing knowledge and to connecting a global community of scholars around biodiversity science. So thank you again for your comments. It's an enormous pleasure to be here today.
Okay, thank you, Professor Davis. Just arising from the comment you made, I listened very well to your presentation. I'm actually particularly happy to know that you mentioned that at least half a dozen of your own plant collections also are not digitized. I thought this silent herbaria is just with Africa or, in the global South. So I'm happy to know that. But do you think that artificial intelligence could be used to perhaps maybe help in the digitization of these plant collections? And if so, how best do you think we can begin to employ that at this level to achieve that? Thank you.
Yes, thank you for your question. I absolutely believe that artificial intelligence can help to facilitate digitization. And so, one of the ways that I think about this and presented in my talk is that digitization is sort of consists of two phases. So 1.0, what we call 1.0, is dealing directly with the physical object. It takes place in museums, in herbaria, with staff who are actively taking images of collections and transcribing labels. Those are kind of the most basic elements of digitization, and we call that digitization 1.0. And from there, we can then make these collections accessible online to the world. So in that case, we're working not with the physical specimen itself, but rather with the digital derivative. And this is what I consider digitization 2.0. And I just want to point out that there's still a lot of innovation that has to take place in the realm of digitization 1.0. There's various collection types that haven't received attention and require novel technologies. And there's just a lot of work to do on making the global meta herbarium or the global meta museum. But once these digital collections are mobilized online, then they become useful for enhanced sorts of digitization. So in this case, not only can we do and practice science that we've always done, but at a much larger scale, but we can apply new sorts of technologies like artificial intelligence. And so here, what I would point to as the most kind of innovative and exciting practices using artificial intelligence are one, transcribing labels. So in the US, we still pay workers to physically transcribe the labels by hand. And this is very straightforward. It's standard practice. But increasingly, computers are able to read these labels and transcribe them automatically. With typewritten texts, that's a much easier proposition. Optical character recognition has been a phenomenon for at least 20 years. And but increasingly, handwritten labels are being able to transcribe from these machine learning models. So that's one way in which we can utilize and leverage artificial intelligence, that is to enhance and speed up transcription. The other way is to kind of harvest and extract biological information from the actual pressed plant itself or fungus, right? So the idea here might be you're training a model to identify features of the plant, to harvest those traits, to score traits from the plants using digitized rather than physical records. So in our case, our lab is focused on crowdsourcing digitized specimens to collect information on when plants are fruiting and flowering and how that's changing with warming temperatures and climate. And then what we're also doing is using these crowdsourced data, these kind of feature or trait data, to train models then to automate the process of phenological scoring. So being able to identify precisely where flowers and fruits and buds occur on those plants and to count those features and then to connect those with climate layer data. So those are two very direct examples of the way that artificial intelligence and machine learning and approaches like that are greatly helping to enhance the realm of digitization and biodiversity science.
Thank you so much, Professor Davis. We'll take on more questions now. Thank you. Okay.
You're welcome. Thank you.
Okay, thank you very much. My name is Lawan Sani from Department of Plant Science and Biotechnology, Bayero University, Kano. Thank you very much, Prof, for this inspiring and eye-opening presentation. It is, in fact, has opened a wide opportunity for us. In my university, we are involved in this silent collection, Iberian collection. This is because many institutions that are around us rely on us on species identification for those staff and student researchers. And we are doing that a lot to make sure that we have, we met up with the demand. What plan is your group having to involve department like us at institutional level? And if we want to join in the digitalization, what steps do we follow to join with, your group? Thank you very much.
You're welcome. Thank you for your question. So I would just, with regard to the previous question, I would just emphasize that species identification is another area where artificial intelligence is likely to be very useful. But training any machine learning model involves high quality, accurate data. And so the identification of plant species really continues to remain foundational for herbarium practice. The identity of species is one of the main kind of functional reasons for herbaria, and that continues to this day. I would say that there are sort of two ways in which we could think about, and what I would also emphasize is that what Daniel and I had identified is increasingly over time and space, there is much more of an ownership of in-country knowledge and in-country expertise in stewarding Nigerian plant collections. And so, you know, this is also something we're seeing elsewhere, but the best knowledge in many cases is likely to be the local knowledge. And this would apply, I believe, similarly to these collections. There are, I believe, two paths forward to thinking about digitizing efforts in country in Nigeria. The first is to work with institutions that are developing this expertise and this knowledge and thinking about efficient ways to make these collections accessible. And I do know that there's collaborations with varying US institutions and those across Africa, including in Nigeria, to mobilize collections and get them online through digitization. So institutions like ours at Harvard and others are already active in those activities, and I would encourage reaching out to those individuals to learn more about those activities and potential partnerships. But I would say, and I always stress, that any record of a collection, any digitized record of a collection is valuable. You know, so even if it's a simple photograph taken from a cell phone, this is much more useful than simply a collection that remains locked behind closed door. The second way I would think about this is trying to really generate local enthusiasm that for this endeavor and this pursuit that actually results in national funding initiatives and efforts to make these collections available online. So in the United States, there's been more than 20 years, arguably now, of the United States National Science Foundation funding efforts to digitize collections. Now, these have been federal sources, but they've also been complemented with internal institutional funding to make this happen. And I realize that's not always possible. It's the same in the United States. But I do know that the US has served as a good model for many European countries now following, with national funding initiatives to make money accessible for digitizing collections and making those collections accessible and available online. Now, this ultimately has to reside with national funding officers and politicians that make this a possibility. But I would argue that demonstrating practice and the value and utility of these collections at the local level and the regional level is important for initiating those efforts. And the work with Daniel and the many other Nigerian scholars and herbarium stewards that were part of the paper that we published are really kind of central to trying to mobilize that energy and that effort, who have already developed this practice and learning from those institutions. And then secondly, trying to work to generate kind of national initiatives that will help towards funding this expertise. And I will just say one final thing is that in the United States, these grants are always collaborative. That is to say, there's never one institution that is the sole funded institution for these projects. These are done in concert by the requirement of the solicitation with a whole variety of institutions. These are small institutions. These are large institutions. These are public institutions. These are private institutions. So all of these institutions, regardless of their size and their expertise, are working together on this common goal, and that knowledge and that expertise is then distributed across this network rather than being solely the responsibility of a single institution. And as these conversations move forward at the national level in Nigeria, I would encourage that model of dispersed involvement and dispersed learning that happens across a range of collections, universities, and institutions be prioritized.
Thank you so much, Professor Davis. Okay.
You're welcome. Thank you.
Professor Falodie has a question for you.
Okay, thank you for the comments and your presentation. Well, I'm going to speak from the perspective of as a natural product chemist. I'm a natural product chemist. In the context of emerging diseases and climate change, what role do you consider could be necessary to revitalize the silent herbarium? I'm saying this because of the issue of chemical ecology that you need to, address when you are trying to address a particular disease condition. In isolating and characterizing chemical constituents from medicinal plants. Thank you.
Yes, that's an excellent question. Thank you very much. So I would say that, as you are aware, much of the world relies on plant-derived medicines, and that's true wherever you are in the world. And I think plants are among the best chemists in the world. And obviously, humans have benefited from that. I would say that it's a very exciting moment for.
Thank you so much, Professor Faludun. Just before I bring on someone to throw more light on what Prof said, I remember when I asked that question, I was thinking, because that's the reality, because we are talking about new frontiers, isn't it, in plant and environmental interaction. So there's already a diversion that we're going beyond the normal. I heard you say, I'm not a botanist, neither am I. I'm actually, I would say I'm someone who deals with the people, who goes to the field, who goes to the bush. I deal with humans as a sociologist, okay? So I deal with humans. Now, but this is quite educating for me. I'm thinking, from what I've heard you say, Professor Iboka, that's like local intelligence. Because I remember that when Professor David was talking, he talked about from the point of using artificial intelligence from labeling. So he didn't talk about going to the bush and getting the identification. Thank you, transcription, that's the word. So which means that someone needs to have gone to the bush, got the plant, and then put it together. I still remember my biology in secondary school. At least when I saw all those things, I was remembering that, in biology that time, I didn't know that was what is called Herborea. Thank you. I learned so much today, pardon me. I'm a sociologist, you know. So I was like, oh, so you mean that's what we were doing? Herbaria, that is what it is. We were taking plants, our biologists take plants and then you label it. And then we'll have like a whole book that comes out and like this plant. I didn't know that's what we were doing. So I'm actually very happy because I've learned a lot today. But I'm just thinking, because we're talking about new frontiers. Artificial intelligence has come to stay. Ethically, how do we go? But I'm thinking, can we incorporate local intelligence, Professor Iboka, and artificial intelligence in doing what we have to do? So we see, because you rightfully mentioned that there's no way artificial intelligence will give you some of those native, which are the silent, the herboras that we have. So if artificial intelligence will not give us native, so we need to have native intelligence mixed with what? That's my line of thinking. But I believe that Dr. Daniel Ziegler will throw more light. Thank you very much.
Hello, everyone. I'm Daniel Ziegler, and I was opportune to visit Professor Charles Davis' lab and Herbaria at Harvard. And so I want to just briefly, we don't have time, to tell us how they operate there, even in the face of the use of AI. They are always ahead of AI, I'm telling you. And they are using herbarium ahead of AI. So what are the criteria for use of herbarium internationally? The first one is to register your herbarium in the global database, which is Index Herbarium. And it's very simple to register. The criteria is number one, if you have 5 to millions of specimens that are allowed, public are allowed to access. If you have only 5 specimens in your herbaria or in your herbarium, you can register it. And then if you have up to 5,000 specimens in your herbarium, you will get funding to mobilize and put them online from IAPT, International Association of Plants Taxonomists. So if you have a herbarium and you have 5 specimens, only 5 as a criteria, you can register. And registration is very simple. Just Google to TIA's Index Herbarium, and then they will ask for your email address, either as a HOD or someone in charge of the herbarium. You give them your email address, and then indicate how many samples do you have in your herbarium. Let's say you have 5. Tell them it's 5. Be specific and be very honest. If you have 5 samples, tell them it's 5. They will not deny you the registration. And once you are recognized, then you are allowed to have access to funding. They will give you like $10,000 to mobilize what you have and put them online. So I will advise the Nigerian herbaria to do this. This is the first basic thing that you can do to register your herbarium online. And then the second step that we can take as a country, most of us here are into academics. They are in the universities. And we have project students, especially postgraduate students. You see a postgraduate student work on a particular group of plants, but he will go to the curator and say, give me voucher number. And the curator ignorantly will go into his book and give you one number that you don't know what it is. So it is encouraged that when you work on a plant, get a specimen, herbarium specimen of that plant, and go and keep it in the herbarium. You are adding to the collection of that herbarium. So by the time you have your own collection in the herbarium, you get a voucher number of that specimen that you collected. It's not the number that is existing in the herbarium that is your voucher number. It's the number, is the specimen that you bring into the herbarium and is kept in that herbarium. Then the curator of that herbarium will give you the number of that specimen. That will be your voucher number. So it is wrong to say, give me voucher number, and the curator will give you a number without a specimen. Whose number did he give you? Nobody can trace. So all of us, if you go back to your institutions, ask your postgraduate students or yourself to collect specimens and put them in the herbarium. That way, our herbarium will grow. The third step, I said 2 steps, right? Register your herbarium. Second, make collection, deliberate collection, and keep them in your herbarium. Herbarium specimens are used in a way that we didn't know before. So the future for herbarium is growing. Most of the branches of botany, they are now sitting in their herbarium and do them. So metabolomics, like he mentioned, you can use herbarium specimen for that without going to the field. You can study pollination biology using herbarium specimen, phenology, the flowering cycle of plants. You can study climate change in your herbarium. So there are so many openings, so many studies that you can do, that you can use herbarium specimen for. So I encourage us to go back and mobilize on what we have and put them in the herbarium. There's an ongoing project from the Savannah. We are mobilizing and digitizing specimens that are deposited in all herbaria in the north. We have finished up to 90%. Most of the herbarium specimens are now on our database. They are on our hard disk. It's not online yet, but that is just a click to say upload. But they are almost ready. And if there is such project in the south, it's complementary. We can put our hands together. The northern part is taken care of, and we are now looking for the southern parts to do their own, so that our specimens will be online. We discussed this at one of our journal club meetings with Professor Charles, and we brought up that project that surveyed most of the herbaria in Nigeria, for which about 50 universities participated. And that was how we discovered that 80% of herbaria in Nigeria are not actually visible. Nobody knows about them. And the 10% that are visible, registered on Index Herbarium, their specimens are not visible on GBIF. Only 5% is available on GBIF that you can download and do your species distribution modeling. So using herbarium specimen alone is enough to do your botany. The entire botany, you can use your herbarium specimen for that. So please, if you have more questions today, now we don't have time, but if you have more questions on how to use herbarium, how to register your herbarium online, please, there's still time during the conference. We can interact, we can chat on way forward. Thank you.
Thank you. Thank you, Dr. Daniel Ziegler. There's nothing as beautiful as having firsthand, hearing firsthand from someone who had that experience with our Professor Charles Davis. We were hearing the Daniel, so we're so glad to know that you were the one that Professor Charles Davis. Please, a round of applause for him again. And you'll agree with me that his comment here has really helped to educate us further on how we can really take advantage of some of these privileges. I'm happy because I see now that this local as well as artificial intelligence is already in view. And if we will take advantage of what this association is presenting to us, the society, Botanical Society of Nigeria, by putting together, let me commend the president again and the members of council for this beautiful, and the LOC chairman for putting this beautiful program together, because you and I would agree that it's not only educative, it's actually providing solutions to some of the things that we have thought before now wasn't possible to be done. We are seeing Dr. Daniel tell us that these things are possible. I'm glad also that we are reconnected back online with our virtual participants. We apologize, our virtual participant, for the power outage, but we're glad that you're reconnected back with us. Just before we were disconnected with you, we were just taking questions. Professor Davis was giving answers to questions and comment that was made by Professor Falodu, and we have progressed a little bit in asking a few questions. But without wasting time, I saw a hand up. I just want to take that one, and then we'll move really fast. Where's the hand, please? That person that raised the hand for a question. Okay, I'd like to know if our online participants also have questions. Please, media, you could take note of that. Perhaps during the plenary session, we would address all of those questions. Is there any question coming forward, or we move quickly to taking the presentation? Okay, if there's no other question, I want to say thank you so much. I believe this aspect of the program has been quite educative, and we have learned so much. We will move now quickly to taking the presentation of our plenary speaker, Dr. Abubakar S. Rigim. So we'll now read his profile. Okay, just been told that we're trying to link him up, but just before that, let's have a comment from our president as regarding maybe some of the comments that have been made so far, perhaps what the Botanical Society of Nigeria will begin to look or do with respect to some of the issues that have been raised. Thank you.
Okay. Thank you. Based on our discussion this afternoon, it is really intriguing, because the issues regarding AI and even our Iberia are kinds. Visibility is really an issue. I've read part of that survey, and it's really unfortunate for our Iberian are not visible. This was raised sometime in a related fora where we discuss about journals and other of our publications. Issue that has to do with being a digital and also operating in another platform. When we are discussing on the old dogma on publish or perish, a new model has come into being, that you be visible or vanish. Now, for us to stand in a global sphere, we have to be visible. We have to keep our records, our collections. Now, but we have to be very careful on how that things are to be done, so as not to jeopardize what we are doing. AI has come to stay. a global trend, and there's no way we can be behind the scene. Harvard has mentioned they are above AI on most of the operations. But it's critical for us to have a policy regarding the use of AI so that our operations must be in tandem with global best practice. Otherwise, we'll be a culprit in our administration of what we are doing. So our communique after this conference, we have to highlight some of these issues. Our visibility in the global sphere, as far as our collections, and as professionals, we should not rely solely on other issues in discharging our responsibility. If we do that, we can really be in the best pedestal. But in any way, we must not compromise our practice. Having use of tools to negate the practice. If that is done, then we really have the issue. And we must go back to basic and build from the basics that we have. Then we get a clear picture of our operations. And I think our discussion this afternoon has opened a lot of things for us as a lesson that we take home so that our operations must all be in line with the expectations. As I've said, as professionals, we must be guided. Many universities, they have policies regarding research, regarding the IPR, regarding this AI. If we embrace all these things, I think we'll never be culprits to our operational mandates. And if that's done, then we get it right in all what we do. Let us be. I want to explore more to Dr. Ziegler on what you are doing on those documentations, to reach out to other universities that are not visible. Let's have a record of our collections and how that can be put in a global sphere so that we'll be one and we cannot hide from what is in there. But we must not forget that the basic tools in taxonomy must to be adhered to as it is collections. We know it is a tedious thing in recent time because of the issue of banditry. Some areas cannot be reached for collections. As we have heard what happened to the vice president, an attempt to reach out. I know he's very good wherever he is. He will always walk around with his camera to make collections as much as he could in any given time. Last year, when we were in Kebbi, he promised to reach out to do some collections. I told him that there are some areas that are not accessible at that time for him to do such collections, that you need to be very careful on where he is to reach. But we thank God, he was able to explore the area and made collections from that end, which I believe will be galvanized into its records and archives. These other members whose herbarium are not listed, I've read that paper, and that many universities, even in the north, were not touched. And I think we share that link for those that have collections. They can easily register. I think minimum of 5 or whatever they are. We have a lot of them rotten. They are not archives. And the trend of things happening with the climate change, there are a lot of resurgence. Some species are taking over and some are completely being phased out. This must be done in earnest to salvage a situation as we are. So thank you once again for being here. I think the discussion is really continuing. We'll continue to embrace voting. So our communique committee, please, let's have the drafts at the end of which we have a take-home on some of these issues that we are discussing this afternoon. Thank you, and God bless us. Thank you.
Thank you very much, President. We will now quickly go to the plenary section. Our speaker is online and ready for us. I'd like to take a brief profile on our speaker. Kindly listen. Dr. Abubakar S. Ringim is a dedicated and accomplished biodiversity conservationist who is deeply passionate about protecting the environment. He holds a BSc in zoology from Bayero University, Kano, an MSc in biodiversity conservation from the University of Dar es Salaam in Tanzania, and a PhD in bioenvironmental sciences from Morgan State University in the USA. He co-founded the Federal University d'Orsay FUD Conservation Society and currently serves as its secretary. Additionally, he is the deputy director of FUD's Center for Arid Zone and Wetlands Ecology. Ringim plays key roles in several conservation organizations, including serving as vice president of the Nigerian Society for Conservation Biology. He is also a member of the Nigerian Coordination Group of Key Biodiversity Areas, a board member of the West African Conservation Network, and a member of the Nigerian Field Society and the West African Ornithological Society. Previously, he was a member of the Botanical and Zoology Societies of Nigeria and is affiliated with the Global Society for Conservative Biology, SCB. In his administrative capacities, Ringim is the chairman of the FUD Zoology Guardian and the deputy coordinator for postgraduate programs in the Department of Animal and Environmental Biology at FUD, among other responsibilities. His research centers on biodiversity, conservation, and citizen science, focusing on translating scientific findings into practical solutions. Ringim has published numerous peer-reviewed articles and online content on biodiversity conservation and has participated in various national and international conferences. Beyond his professional work, he is an adventurous nature photographer who uses his passion to inspire others in the field of conservation. Ladies and gentlemen, a round of applause for our plenary speaker, Dr. Abubakar S. Ringim.
Thank you very much for the wonderful introduction. Some great discussions going on. I haven't had some of these great discussions in a while. Thank you, everyone, and it is a great pleasure to be with you today. And I am particularly interested. All right, thank you.
Thank you. We're back on.
What about now? Is my screen visible?
It's showing now.
Yes, it's showing. We can hear
Okay,
you. We
great.
can see
Thank
you.
you. All right, thank you. Like I said earlier, such are great discussions, and of course, some of those questions and some concerns raised will be addressed or will be of importance in my presentation. And of course, my presentation will add light and value to some of these discussions we are having today. So the title of my presentation today is New Frontiers in Phytogeography, Unveiling Plant-Environment Interactions Through iNaturalist in Nigeria. And of course, as we had the word from the title, iNaturalist, some of this here have been using iNaturalist for a while. Well, some are hearing it for the first time. I would like to begin my presentation with this important quote. We may have our differences, but that is one thing we all have in common, which is Earth, and we have to do everything to defend it. So an iNaturalist is a social network that is conservation focused. And what or how it works is for someone to record their observations. You go to the field, you see something, plant, insect, mammal, or whatever you see, any critter, you just take the picture and you share it online. When you share it online, there are other fellow naturalists and citizen scientists who can help you identify what you observed. And of course, some of these findings and recordings add or generate a lot of great discussions. And of course, some of these pictures that you see in the wild turn out to be new species, as we'll see in the presentation. And of course, if you are not a fan of taking pictures, you can help identify what other citizen scientists are uploading on the iNaturalist platform. Of course, we talk about taxonomy, taxonomists, there are a lot of issues going on with the taxonomic studies. So this is where taxonomists come into play. This is where they really add value, especially when we talk of the flora of Nigeria, where we are talking that less than 10% of the Nigerian flora is digitized. And we have a lot of insufficient taxonomies in the country. So this is where they come into play. So what's the origin of iNaturalist? It began as a master's project at the UC Berkeley School of Information. And of course, it differentiates from other traditional citizen science community science as it is not top-down or directed. With regard to traditional citizen science projects where scientists involved the public to collect data on their behalf. And of course, such traditions, while it provides opportunities for the public to engage in science, we believe that traditional citizen science projects lack the ability to foster deep, long-term investment in the natural world because the participants don't have a strong connection with the science. However, compared to iNaturalist, it focused on facilitating a lifestyle of data collection because the scientists, the public, the citizen science go out to the field and collect this data himself out of passion. And this encourages data contribution through the self-interest of an avid hobbyist, not the altruistic desire to assist in someone else's work. Because when someone asks you or pay you to collect data on his behalf, you are doing it, you may not really do it out of passion because you are doing it for the sake of someone who pay you to collect that data. So basically, iNaturalist does that by focusing on user experience. What the users, what the citizen science feel, how do they feel, and how do they contribute to this data to biodiversity conservation? And also, how do they value this observation? So it is openness in both community and data because citizen science data is freely accessible by everyone and by encouraging civil evidence-based discussion and identification. Because when you take a picture of a plant and you upload it on iNaturalist, it is available and visible to all people online. So a lot of people will add great discussion, identification, and information about that particular observation, be it plant or whatever it is. So it's really a community that shares, discusses, and identifies their observation of wild organisms. So of course, we talk of wild organisms. On iNaturalist, you can still upload the records of introduced organisms, like invasive species and so on. And by doing so, generate scientifically usable data because you share, you discuss, and you help identify. So with over 500 citations, iNaturalist is the most cited data set of the over 60,000 tracked by the Global Biodiversity Information Facility. It is the largest digitized database that we have today in the world. So sometimes when you take a picture of this species in the field, they start a great discussion because every observation is someone saying, I saw this thing at this time, at the place, and what do you think? Because whatever record you upload on iNaturals, it has three key items or things. One, it has a location, it has time, and it has the record of the person who uploaded that data or record on iNaturals. So it helps us track when, where, and who submit or upload an observation wherever in the world. So how does an hour look on iNaturalist? So this is from October 2021, an hour on iNaturalist. You can see the flow of submissions of observations from different parts of the world. So in an hour, over 5,000 new observations were submitted, uploaded on iNaturalist. And out of this, people or other citizen scientists added over 12,000 identifications. Some of these are scientists, some of these are naturalists, some of these are taxonomists, some of these are just citizen science helping provide ID to observations that people upload on iNaturalist. And within an hour, there are over 200 new participants, people joining iNaturalist platform or database. Some of these discussions can get pretty involved and pretty nerdy because of the of their interest in particular. Any other record is valuable and is of interest because it helps scientists track biodiversity distribution and occurrence across the world. But yet some of these are more valuable than others, especially when you talk of threatened or rare species. You can see some of these people have provided their some of the information they know or ID or bit about this particular records. So it's kind of an online database that provides an avenue for scientific community to interact, discuss, and share their findings or observation in the natural environment. We can simply say that it's like a virtual herbarium or a virtual museum if you talk from the fauna point of view. So currently, this map was generated 2 days ago. So currently, there are over 288 million observations, individual observations of flora and fauna on iNaturalist database. And this comprises over 516,000 plant and animal species. So like I said, iNaturalist is the largest database of plant and animals in the world. And a lot of scientists, a lot of protected area managers, a lot of people, scientists are utilizing this observation to influence decision and policy regarding biodiversity conservation. iNaturalist is by far the largest source of most species on JIP. You can see, compared to other online databases that we have, the Cornell Lab of Ornithology and other large publishers, you see iNaturalist over 400,000 citations. So sometimes it is not just about the data, the record you upload. Sometimes it is about the passion, the fun you have using iNaturalist. And this guy is saying that iNaturalist has become a part of my daily life. It helps me learn about and keep track of my local biodiversity because you can actually create your own life checklist of biodiversity. When you go to the field, we take a lot of pictures. Like for me, for example, I take pictures of everything I see in the field, be it plants, be it animal, be it insect, whatever. I just take a picture of that because it helps me create my own lifetime database. It is like a Pokemon, but really it's become my obsession, especially during the pandemic. I naturally don't know. So that's some of the discussion, some of the experience people have about using iNaturalist. And of course, you can do it from your backyard. You don't have to travel far to go to a protected area, a reserve, or a national park, or a botanic garden. You can simply do it from your backyard. What you see, you just take the picture of it and you upload. And of course, that adds value to wildlife conservation. So how are we doing in Nigeria? We have seen that in the world, there are close to 300 million observation pictures of flora and fauna, including their information related to where and when they were seen and who last uploaded these species. So with regards to Nigeria, with regard to iNaturalist usage in Nigeria, we are doing very good. So you can see the graph from 2019 to 2024, you can see how we are doing observations per month. So the observations keep increasing. And of course, we will see in the next slides how many people are contributing this data to Nigerian, to mapping biodiversity occurrence, flora and fauna in Nigeria, which is very exciting and it's very encouraging. Yeah. So currently, there are over 2,670 plant and animal species uploaded on iNaturalist from Nigeria. And you can see the geographical distribution or occurrence of this data. You can see where observations are concentrated, and you can see some areas where there is sparse data. And of course, we know some of the reasons why these areas are poorly surveyed is due to insurgency and security concerns. And it is interesting to see that less than 2,000 Nigerians contributed this data on iNaturalist. And of course, we know our population is over 230 or 40 million. And out of that, less than 2,000 Nigerians are contributing, creating this remarkable data on iNaturalist. So some of the interesting and fascinating observations over the years of iNaturalist in Nigeria included the first ever living photo of Dicostatoides beetle, which was observed by a friend, B.B. Tayo. And this, you can see some of the comments by some of the iNaturalists, because on iNaturalist, there are people who are very famous, who are authorities when it comes to all tags. So they keep track of some of these organisms, using a particular or specific project. You can read more about this observation. It is available online. And of course, another interesting observation is the yellow-winged bat. And this is the second record of this from Nigeria. The first record was uploaded on iNaturalist in 2021 during one of our visits to Kainji Lake National Park. And this appears to be the second record of it from Chad Basin National Park in Tabona. And a third record was uploaded on iNaturalist 3 months ago by a friend. Also, apart from mapping the observations of key species, there are, of course, another interesting things that happens on iNaturalist. This is an African silver-billed bird that was observed using a weaver bird nest. And we know that silver bills do not lay eggs or do not utilize the nest. But of course, this is one interesting thing and about iNaturalist, because it helps you understand species ecology as to what they use, their habitat use and patterns, for example. And also, of course, if you use iNaturalist, there is a lot of great deal you can use with the data you generate on iNaturalist. And this is the first up-to-date biodiversity inventory in Hadeji Wetlands National Park. And this is out of one of our ongoing projects in Nigeria. We were able to publish the first up-to-date biodiversity inventory of this national park. And this national park was recently upgraded from game reserve to national park. And of course, this survey that we conducted will provide a foundation for the management of this important national park. because there has never been an updated and comprehensive inventory of this national park. Most of the studies being conducted and field surveys were on specific taxa, but in this study, in this survey or biodiversity inventory, we surveyed all creatures, all plant and animals, including the understudied taxa in this national park. We recorded close to 400 species of plants and animals. In fact, for birds alone, we recorded over 280 bird species over 11 months. This was published recently. So what's like look like the citizens' view of plant environment interaction in Nigeria with regards to iNaturalist? So currently, we were able to map or upload 1,856 species of plants from Nigeria. So, and it is interesting to know that less than 2,000 Nigerians uploaded these records on iNaturalist. You can see specifically 1,882 observers uploaded or contributed these observations on iNaturalist. You can imagine if 10,000 Nigerians or 20,000 or 50,000 or 100,000 Nigerians out of the 240 million Nigerians will be involved in this platform, you know? So it is kind of very interesting and remarkable efforts from very few Nigerians who go out to the field, take pictures of plants and upload them on iNaturalist. And you can see the occurrence of these plant observations, mostly in the far north and also in the south, with parts of north west, parts of Sokoto, you know, poorly surveyed, and also parts of Borno poorly surveyed due to, of course, security concerns, among other reasons. Also, fewer citizen scientists who go out to the field, take pictures and upload on iNaturalist. So what can we deduce from this observation? Let's look at some of the interesting things from some of this information available on iNaturalist from Nigeria. So these are some of the most observed plant species. Out of this 1,856 species, you can see some of the most observed species. The most observed plant species is neem, Azadirachta indica, and the second most observed species is African mahogany, Caya senegalensis, and then of course Indian mango, Kashmir tree, and so on. So these are the mostly observed plant species on iNaturalist from Nigeria. And of course, we can take a look at the number of introduced species in Nigeria. So you can be able to filter, are these wild species, are they introduced, or are they native species? So currently, based on the data we have on iNaturalist, there are 91 species of introduced species in Nigeria. Of course, if you look at other database available or herbarium records or other things, of course, there could be more than this number. But based on the, speaking from iNaturalist point of view, the number of introduced species that we have in Nigeria are 91 species. And another interesting thing to see or to know is that not only mentioning the specific species that are found, it helps you track their occurrences, their distribution, as also their abundances in terms of number of pictures uploaded. For example, for neem, you can see that 200 observations. For Indian mango, there are 144 observations. For teak, there are 101 observations. This gives you an idea of the occurrences of these introduced species in Nigeria. And let's see their geographical occurrences. You can see that this is the geographical occurrence of introduced species in Nigeria, the 91 species, and you can see where they were mostly observed. And this can give you an idea of where these introduced species are found, and that will help you in managing them. And like I said earlier, each specific record, each specific picture uploaded by iNaturalist has three key elements. One, it has where it was observed, uploaded. And secondly, it has time. And thirdly, it has the record, the name and details of the observer. And that gives you an idea to make a follow-up with regard to that observation. For example, if you are a scientist and you are interested in a specific record. So with regards to number of threatened species, how many are there in Nigeria? Of course, if you check other databases, we know that if you check, for example, the IUCN Red List of threatened species, they are far greater than this number of threatened species in Nigeria. But of course, this based on the available information we have on iNatural's database, we have 327 observations of 49 species of threatened plant species in Nigeria. And also, you can be able to track their abundances in terms of the number of pictures submitted, uploaded on iNaturals. So you can see African mahogany, it has 155 observations. She3, it has 35 observations. You can be able to tell, okay, among the threatened species, which one has lower occurrences, and that will give you an idea of its maybe rarity, for example. So these are some of the things that that have been digitized and are available to us. And of course, you can also take a look at the geographical occurrences of these threatened species in Nigeria. You can see the place where mostly they are observed, parts of Ibadan, and also in areas around Bauchi. Of course, that's the area around Kainji, and also in Katsina, because there is an ongoing project by people in Katsina about the flora of Katsina. So they are doing a great job in mapping the flora in Katsina. So that adds to these observations. So we have seen some of this data, and what does this tell us? Of course, it helps us identify conservation hotspots. The data can reveal areas with a high concentration of species or unique plant life that may warrant protection, especially if we are talking about endangered species or threatened species or endemic species, for example. Also, the data help us monitor introduced species. Observation can provide early warnings about the arrival and spread of invasive plant species, critical for rapid response and management before the species become widespread. And in the picture, you can see the geographical occurrence of Chromolena odorata in Nigeria based on iNatural's database. So you can see the geographical occurrence of this plant is largely limited to southern parts of Nigeria, with a few records in the north, in parts of Gashaka, Gumti, and in parts of Abuja. So this gives us an idea of how we can tackle some of these introduced species in Nigeria. So it helps a great deal in management of invasive species. It also helps us assess species range and status, because we are talking about climate change, we are talking about habitat degradation, a lot of impacts, a lot of human activities that impact wildlife, and of course, the distribution of flora and fauna. So some of this data on iNaturals can help us track a range of species, because the data will help us in providing, for rare and threatened species, new observation can provide crucial information on their population health and potential range shift due to climate change or habitat loss. So this is yellow trumpet Costus spectabilis, which is the Nigerian national plant. Based on the iNaturalist database, you can see the geographical occurrence of this plant. It was mostly observed around Gashaka Gumti, which is mountainous area, and also parts of north central, parts of Bauchi, where the Ningi bush and outcrops, which is also mountainous, and of course a few places around Zaria. So this help us track the geographical occurrence of some of these rare species that we have in Nigeria based on their frequencies of occurrence in the field. Also, the information available on iNatural's database can help us detect environmental change, because we can compare the observations over time. We can do an assessment of the in changes of floral composition or abundance, for example, from what we know in the history to date, or we can even project in the next 50 years what will happen based on current projection for climate and environmental variables or human impacts, for example. Also, a decline in certain sensitive rare species or a change in plant community can signal environmental stress. And of course, some of this data are proving to be very valuable in tracking the geographical distribution of plants and animals. Another important thing that we can do with this data is to be able to influence policy and decision making, because the verifiable data on iNaturalist can be used to support conservation proposals. inform governmental bodies, and provide a rich data set for further academic research into plant ecology and distribution. Of course, we are talking, our keynote speaker talked a lot about plant phenology. You know, a lot of plants due to climate change are changing their time of fruiting or their time of flowering and things like that. So this data available on iNatural is already proven to be very valuable in tracking the phenology of plants. In the picture is American throat lily. You can see in 2016 through 2017, it was observed the phenology of this plant was in April 2016, 2017. But then compared to 2014, 2015, it was mostly in May. So with this data available to us, we can be able to track the phenology of plants in Nigeria. Of course, whatever is good has its challenges, and we can see some of these challenges: constant tension to trying to cater to an audience that ranges from amateurs to world experts, especially when it comes to Nigerian flora and fauna, for example. We want a lot of people, a lot of experts sitting with us today to join iNaturalist platform so that they can be able to add to their expertise to understanding the Nigerian flora. Of course, we can have people who have expertise in Nigerian flora from other parts of the world, or people who have immense knowledge about flora of the world. But of course, not like our indigenous experts who locally know this native species. So we really want this expert to be available on iNaturalist database to provide accurate identification to most of our observations from Nigeria. More users who can help identify observations are always needed, especially on the served areas and taxa, which is generally speaking about plants and animals. Taxonomic disagreements, a lot has been about this.
It amplifies the sound wave out from the mic, so that all this strong.
And also making data open for research and conservation, but making sure it is not being used for nefarious purposes. Like I said, citizen science data is mostly available to the public, but of course we need to make sure always that this data is not abused. It's not abused. It is yet another social network someone has to join. We talk about other social networks like Facebook, for example, like Instagram. Yet, iNaturalist is another social network, yet very fascinating and interesting when it comes to scientific studies. There is general lack of enthusiasm. So there is also a lack of enthusiasm. A lot of people are not get involved, and sometimes it is very difficult to keep people involved due to lack of enthusiasm. So what are the key takeaways in this presentation? iNaturalist is indeed advancing phytogeography by making a significant contribution to understanding and conserving plant diversity in Nigeria. We have seen how iNaturalist is shaping our understanding of the geographical occurrence of threatened species, introduced species, and other native and wild species. It is interesting to know that over 20,000 photos of more than 1,800 plant species have been uploaded to iNaturalist from Nigeria in just 12 years. And remarkably, less than 2,000 citizen scientists in Nigeria have taken part in this effort. Nigerians are on the way to creating the first comprehensive checklist digitized of the flora of Nigeria. And of course, we know that compared to other countries, they have produced their own flora, checklist of flora. And of course, Professor Abokan has done a great job with regards to checklist of vascular plants in southern Nigeria. Yet, we need more of them to be able to produce our own red data book and also our own national flora. And of course, in other various biological taxa that we have in Nigeria. So what are the future directions? Expanding and enhancing INAT, citizen science engagement, targeted INAT project to address critical gaps. We have seen that in Nigeria, there are some critical gaps in some areas due to lack of citizen scientists or poor iNaturalist biodiversity mapping activities. And deepening ecological and phytogeographical analysis on plant-environment interaction. This is particularly true when we talk with regard to understanding the impacts of climate change and habitat degradation on Nigerian flora. A lot has been discussed today. Also, we need to utilize this data and also utilize species distribution model to understand change in the geography of Nigerian flora. A lot of environmental threats are ongoing in Nigeria due to high environmental degradation, pollution. Our own population is contributing significantly to environmental change. So we need to utilize this data to be able to tell a story about the change in the Nigerian flora over time. And of course, another important thing is to translate this data into actionable conservation strategies, not to be producing papers, scientific papers. We need to see action. We need to see realities. We need to increase collaboration among different stakeholders, politicians, decision makers, protected area managers, scientists, public, indigenous people. We need to also prioritize our conservation areas. And also, this data can also improve monitoring of Nigerian flora. And of course, sign up with iNatural is free and fun. And of course, that will make a difference in biodiversity conservation. Thank you.
Thank you so much, Dr. Abubakar Regim, for your very insightful presentation. I believe there are questions that will be arising, of course, as you have asked. Any questions?
Hello, is the organizing there?
Yes. Can you hear me? Dr. Abubakar, can you hear me? Dr. Abubakar, can you hear me? Hello, Dr. Abubakar. Guess it's a slight. Okay. Hello, sir. Thank you so much. Can you hear me? Wave if you can hear me. Oh, I guess. Please see. Okay. Can you hear me, sir? Dr. Abubakar.
Yes,
oh, beautiful. So we're saying thank you so much for your very insightful lecture. We're happy to engage with you. Thank you so much for speaking to us. I believe indeed there will be questions. We're moving straight into the plenary sessions that will be headed by Professor Aboka, where the questions will come up. So I believe yourself, even Professor Davis and others on the high table, Professor Esther, Professor Falodu, and the President will all be engaging us in this session as we go on. Thank you so much for the presentation. It's been quite intellectual and engaging as well. Thank you so much.
Thank you, Dr. Ringim.
Thank you.
I think you are the current president of Society of Conservation Biology, right?
Yeah, by October.
Yes, by October, okay. Dr. Ringim has been doing a fantastic job with this. I first got introduced to iNaturalist through him, and I've been struggling to get used to it. I recall that I gave a student, an undergraduate student, a topic on iNaturalist last semester. And the student came to me and said, this is not scientific. And she wanted a change of topic. As is usual with me, I told the student that science or not, go and do it. But after she got into it, she now became an apostle for iNaturalist. So it is very interesting for every one of us to be involved in this. We are all potential contributors to fill in the gap for Nigeria in terms of the number of biological species we can show the world that are found in Nigeria.
Yes.
So please get your phones ready. Any plant you get, just register with iNaturalist. It's free of charge. Maintain an account and upload your pictures as they come. And you don't even have to identify it. You just, some people online will identify it for you and help you with the identification. iNaturalist has a way of suggesting names for you on what you have uploaded. So it's a very good interactive program. So thank you very much, Ringim. And I think for this your project, you need to recruit more participants. Although I don't think you are an agent of iNaturalist, but being an advocate for it, what is the likely best method to have people, scientists or non-scientists alike, to buy into this project and advance it from the level that it is right now? Thank you.
Okay, thank you very much, Prof, for the comments and also for that interesting question. Yeah, of course, like I said, one of the greatest challenge is lack of enthusiasm, the ability to recruit and keep people involved in citizen science project. Because one thing about citizen science project is that it is a non-paid work project. You are not being paid for uploading pictures on iNaturalist. It is something you do out of passion, And of course, all of us sitting here today, if you don't like plants, you like insects. If you don't like insects, you like mammals. If you don't like mammals, you like reptiles or birds, for example. So you can just pick a specific area, be it reptiles, insect, plants, and you just contribute to your observation to mapping Nigerian flora and fauna. So, but I believe we can do a lot of great deal of involvement if we keep speaking iNaturalist and the potentials of iNaturalist in mapping Nigerian flora and fauna. And sometimes it's very sad to see that a lot of countries have gone far in creating their own red data book and comprehensive checklist of their flora and fauna, but we are very far behind in Nigeria. So that's the way forward, I think, online webinars, workshops, trainings, and of course, some citizen scientists have to realize the potential in utilizing iNaturalist data. You can create a live list with them, and also you can create a checklist of your protected area or a particular habitat. In fact, you can even create a checklist of your university. And with that, you can be able to tell some stories about this. So the citizen scientists, the public need to see value in what they do. So that's one key way I think that will keep people involved in this kind of projects. Not only iNaturalist, generally citizen science projects.
Thank you. Any other questions or contribution? Yes, please. Okay, Daniel.
Okay. I've been a fan of iNaturalist since 2017, but largely in South Africa. So I like it so much because it's a network of experts where if you upload your image, people comment on it, and you get a very reliable information about the biodiversity data that you are handling. So I'm very excited about the use of AI. And one of the major challenges that we faced, or they face in South Africa, is they have some plants that are rare or critically endangered. So if as a citizen scientist, you take an image of this picture of this plant and upload on iNaturalist, poachers will have the knowledge or will know the specific point, coordinates of where you uploaded the image, and they'll go and harvest it for sale. So those critically endangered species were more threatened by people who are on iNaturalist harvesting those plants. For Nigeria, Costus spectabilis, for example, our national plant, is one of the threatened plants that we have, and you don't even see it anyhow. I saw and collected one in Gombe in 2020. When I went back in 2022, it was not there. My problem or my challenge is, what would you advise us on poachers that use this iNaturalist to go and harvest the very few plants that we have? Do you still advise us on the use or uploading those positions or location of those plants. In South Africa, they have regulations in place that you cannot collect plants anyhow, because if the authorities will catch you with a piece of plant, they will collect you, they will ask you if you have a collection permit. In Nigeria, we don't have anything like this. You go and collect plants anywhere and use it for anything that you want without anybody harassing you. So what can you tell us if you join iNaturalist about uploading our plants that are critically endangered? Thank you.
Thank you.
Thank you. Yeah, thank you very much, Daniel, for that great question. Yeah, of course, that has been a great concern with most people using iNaturals. And of course, something has been done on that. I'm not sure whether you are aware. So all coordinates of threatened species uploaded on iNaturalist is now obscured. You can't see the coordinates. Even if you see the coordinates, it's not a true coordinate. So if you are dealing with a threatened species, for example, let's say I'm interested in understanding the geographical distribution, the distribution of flora in Nigeria, and I want to embark on a PhD research, for example. So if I want those coordinates of the threatened species, I have to write to iNaturalist that I want the real coordinates of this species, because I'm writing a project or I'm undertaking a thesis, this, and that. So that has been taken note, because we are dealing with some species are highly endangered. So we can't risk uploading their picture, including their coordinates. So that has been taken, that has been treated by iNaturalist, and I hope that helps. Yeah.
Okay, just as a follow-up to Dr. Daniel's question, sir, because I'm thinking, is that possibly the reason for poachers? Is that possibly why we have some of these herbarium being silent? These native plants, is that why?
No. I think the silence herbarium is just our inability to put our specimens online. It's not about rarity or threatened status of our species. And that is the beauty of herbarium. They keep those records in place. So instead of going to harvest plants again, you already have them in the herbarium. So for research purposes, you can use herbarium specimen for whatever research you want to do without going back to the field to harvest those plants. And as a plant collector, I remember going to the field with Professor Ibukan, and we saw this Costus spectabilis, and it was only one individual. I told him I would plug this plant. He said, no, it's only one. But I told him I don't have record of this plant, and I need it. He says, better you leave it so that it will continue to be there, and take images, or you take a piece for DNA material. Just take a very small piece and dry it for your studies. But you cannot unseat a plant that you know is very rare as a taxonomist or as a botanist. So you go back to the herbarium and use what is available in the herbarium instead of using what you know will go extinct.
At this point, can we put our hands together for Professor Ibuka for not unseating that plant? That's him being very ethical, even in his practice. Yes, I like that. Thank you so much, sir. But again, I want to follow up with one question to you, Doctor. Permits me, I said earlier on I'm a sociologist, yes. In the earlier presentation by Professor Charles Davis, he noted that we had over 500 plants that were not digitalized. As a matter of fact, only 10%. But I was surprised listening to Dr. Abubakar, and he talked about the fact that we had over 1,000 or how many of such plants that are digitized. So I'm wondering, is it that the data with iNaturalist was not available to you when you and Professor Davis was going about that research that made you to say that your findings show that we had over just 5 or so? I need some clarity with that. Thank you.
I think they are different. The data on iNaturalist are observations. They are not physical specimens. The one we used are the materials that we have in our rooms called herbarium. Okay, so 10%, only 10% of our herbaria are registered online. 5% are visible on GBIF. By that I mean, if you Google Nigeria on GBIF, you see our specimen. You can see specimen from UNN. You can see a few specimens from ABU and Lagos. But any other herbarium in Nigeria is not available. So that represents only 5% of our own specimens that are in our herbaria online.
Yeah.
So the iNaturalists are just observations. They are not physical specimens. And it's important to upload or to have observations online. That's okay. Because it does not involve you harvesting the plants. Okay, so there are two different online.
Thank you so much for the clarification.
Okay,
I'm learning. Thank you.
Any other? Okay, just before you, that's Dr. Gumi, right? Yeah. Okay, before you, I just want to take note about the question Daniel asked. Now, you find that, I would say, 30 to 50% of the plants we have here have not been documented in terms of their conservation status. In which case, even the iNaturalist does not know that they are endangered, and they might be endangered, because we have not really done our basic data assessment. So how does iNaturalist deal with a potential endangered plant that has not been so categorized at the IUCN database?
Okay, yeah, so that's a great question, Prof. Yeah, so basically the way iNaturalists categorize whether a plant is threatened or not, because in Nigeria we lack a red data book. So compared to other countries, for example, like Kenya and South Africa, they have a red data book that, for example, if they categorize a plant A as endangered, on IUCN status, it can be categorized as vulnerable or even not endangered, for example. But in their country, they have a red data book that they categorize this plant as endangered. So we lack that in Nigeria. So iNaturalist uses the global IUCN database to categorize a Nigerian plant as endangered or not. For example, if on the global database, Kaya senegalensis is vulnerable. So if Kaya senegalensis occurs in Nigeria, iNaturalist will categorize that plant as endangered, for example. But in the absence of those plants that they are rare to Nigeria, or for example, endemic, for instance, iNaturalists don't know whether these plants are locally facing extinction or not, whether to categorize them in one of the conservation statuses or not. And that's the importance of having our own red data book in Nigeria. Because a certain plant A can be highly endangered in Nigeria facing extinction, whereas in the other parts of the world is least concerned. It's not facing extinction at all. So that's the importance of having our own red data book in Nigeria.
Thank you.
Thank you. Yes, Dr. Gumi.
Yes, thank you. And thank you, Dr. Ringim, for this presentation. I just want to make a little bit of contribution towards these two presentations that we had this morning, which mostly centered on how technology, mostly AI, can be used to enhance conservation and classification of plant species. We already knew how herbarium is central to plant research, whether you are into physiology, ecology, proper identification is the key. So the use of the word digitalization, as Charles rightly said, is mostly to use basic knowledge of taxonomy to identify your own plant species, categorize them, then make them visible. Digitalization is just to make them visible. Just like the olden method we are using when someone published an article and it remains in paper-based journal lying in our libraries. When someone's in 80s or 70s want to use a journal, they will ask you to go to University of Ibadan. It's the only library you have journal there. But with this AI, I believe as botanists, we should all be happy that it's a new technology that will enhance our participation in taxonomy. We have seen how experienced botanists here have mentioned how they view taxonomy as a daunting task. When you look at a university in our own, in UI, you see the taxonomists are somehow looked into that they are just people that roam around the bush making collection. But when you make a field of science more interesting, participation will increase. Younger ones that are coming will know, yes, this is an interesting field of study. You use sophisticated materials. Before you use only, plant press, not even cameras we use. Now we are using high-resolution cameras. You use adhesive to plants, to stick plants, and lots more. Even kids now will be passionate about studying plants, herbarium, and other things. So I think AI is a good technology that can be used. And I call on this society, our project of flora that has long been going up and down. With this, it's a new opening, either with iNaturalist or whatever, we can initiate taxonomy of flora of Nigeria and make it more visible to researchers across Nigeria and even beyond. Thank you.
Yeah. Thank you very much, Lecter, for that insightful
Chairman, sir, thank you for the opportunity. But there's a question in relation to I think some of the comments he made. This person is asking, in light of the presentations, I think the keynote present, will there be training organized for educators, curators on herbarium, digitalization? When can it be scheduled, if any, this year or next? So somebody is interested, we want to learn about the act. I think as we're trying to encourage young ones to be involved in taxonomy. Is that what you call it? Thank you. Okay, so more questions, please. Okay, thank you.
Thank you very much. Maybe I need to apologize that I have the tendency of speaking at times like a critic. That's my disposition. So maybe I need to apologize on that. Ogbuefi, Prof. Ralph Okibo, you said Econia crassipis. So if you Google now, they now call Pontederia. Professor Folorunsho from University of Ife is on sabbatical in my own university, Adekunle Ajashi University, Akoko in Ondo State. Our Vice Chancellor also happens to be a botanist, a plant scientist. He graduated first degree, University of Benin, then later on, University of Ibadan, PhD. But that one is not the central issue. What I'm trying to say is that Professor Folorunsho said while he went to China in 2017 that they have this body, International Association of Plant Taxonomists. They are the ones in charge of naming and renaming, changing and all what have you. Then he also talked about Chromolina odorata. I suppose to me, in our own area, it seems the Chromolina is gradually being replaced with the Mexican sunflower. It's just recently that I learned that it's highly medicinal if you take the infusion for malaria, fever. Talking about my VC as a botanist, I'm conscious that the Vice Chancellor here is also a botanist. He taught us, maybe I should say Professor Raf, Professor Aigbanka, myself, I wouldn't know whether there are others here too. But some of us, like I'm a member of the council, a middle-level member. My organs are on the high table there. So when I read the WhatsApp, I learned that we are to meet with the prof by 9 this morning. I was happy that it would be a forum for me to interact with him once again. So I was anxious that, okay, if I miss that one, maybe I will even meet him here. But that one is just a small issue. But the last point I just want to make is that when we had Professor Oluwatoyin Ogundipe as a taxonomist biosystematist, he helped us to shoulder this concept of having a flora of Nigeria. But the whole thing eventually became like a dead horse. Thank you very much.
Yeah. Thank you very much,
Thank you for your comments.
Yeah, thank you very much. Like I said, in some of the challenges is taxonomic disagreements. Sometimes you see a plant species is named this on iNaturalist. If you check IUCN database, you see it is named this. But mostly that has to do with the common name, the English name. But of course, the scientific name should remain the same. So that's taxonomic disagreements among world taxonomists. Thank you for that comments and contributions.
Next question.
Not really. Not question. My name is Dr. Muda Shiru from FUNAB. Please, we want to use this conference to create an avenue to establish flora of Nigeria, because many of our indigenous plants are listed in the FWTA, Flora of West Tropical Africa. And here in Nigeria, we should have flora of Nigeria. As a plant taxonomist from Southwest, I would like to work with the plant taxonomy in the North, because I have some colleagues there, particularly Abusaria and Sokoto State, and also about the, sorry, okay, thank you. The plants Dr. Daniel mentioned, we have more than 1,500 plants digitized on GBRF, but those plants coded with FHI, UI, plant species. Plants from the Unilag Herbarium, also part, University of Ibadan, UIH, but many are from FHI. And Abusaria plant herbarium is also part. So let us establish the flora of Nigeria, and let us have symposium, both northerner and the southerner plant taxonomists. Thank you.
Okay, thank you. He's making a proposition, I believe. Do you want to respond to that, Dr. Abubakar?
Yeah, I think that's a very good move, and in fact, I will say that that's what I will challenge this society today. They should try to come up with the flora of Nigeria, and of course, one of the easiest way to go with that is to have citizen scientists involved, because creating a flora of Nigeria is a daunting task, and I believe that's one of the reasons why the committee that was set up earlier died, because it's something that needs time, it's something that needs resources, and it's something that needs human power in terms of people who will go out there and create and collect the data. So if a symposia is held, then modalities can be outlined as to how to go about that. And of course, the way I see it compared to other projects that have been held in other parts of the world, so you create an online iNaturalist project called the Flora of Nigeria. Then most of us are teaching in universities, and some of us are researchers in other research institutions. So we form like plant conservation clubs where we recruit undergraduate students. They join these clubs, and they go out on monthly basis or at the end of every week on weekends to just take pictures of plants and upload on that specific project, the Flora of Nigeria, on iNaturalist database. So if we have this kind of project, in some states we have 2 universities, 3 universities. So you can imagine the kind of data that will be uploaded from Nigeria in every month. from different parts of Nigeria. We are talking about 36 states. So if you can have like 1,000 students or 2,000 students or 3,000 students join that project, I believe in 10 years' time or in even 5 years' time, we will have a draft of the flora of Nigeria. So this is the way I see it, because it is a daunting task. And of course, those people that are going to be involved doesn't have to be taxonomists. Of course, the taxonomists are the upper part of it, because they are the ones who know the know-how about the Nigerian taxonomy. But those at the bottom should be like field collectors, mostly students, so they can do this out of passion. And of course, people like me that go out that are very passionate about field work, we do a lot of plant collection. So I believe we can go a long way in creating the flora of Nigeria.
Okay, thank you, Dr. Abubakar, for supporting the motion also raised by one of us here. I just want to quickly remind us that this is a plenary session being chaired by Professor Emmanuel Abubakar, and we also have other distinguished personalities on the high table to also answer our questions. So not necessarily all the questions going to be directed to Dr. Abubakar. I know he had just given the plenary speech, but then I want us to also recognize that we also have other persons. Even Professor Davis is supposed to be online as part of the plenary. We also have Professor Esther Adelawa as part of the... Yes, and our own distinguished president, who's also part of the plenary session. So please make sure that your questions are well directed.
Yes, I think... you want to speak? Okay, as we are waiting for the conclusion of this session, I would like to congratulate Dr. Ringin for this very interesting talk. And for the information of the audience, Dr. Ringim is actually a bird specialist. He works with birds. What do you call it again? What's the name for birds? Ornithologist. Yes. He's an alumnus, or probably an alumnus of Aplori. And to have so lucidly presented this talk on plant related shows how versatile he is. So if we were talking about birds now, I wonder the level in which he will take it. So in appreciation, can we just give him a clap offering? Yes. Thank you very much. So
thank you very much.
We would like to conclude this session now and to go into the next session. The day is fast spent, and I want to call on some people have identified. Okay, we need to introduce to the audience the copies of the journals that are available for distribution. So he will take charge of the process. Is Professor Ajibade around? While we wait for Professor Ajibade, let's take a few comments.
I want to thank my close friend, Dr. I'll remember now, who just corrected me now. You see, I'm not a taxonomist. I didn't even know that. I was so impressed that when he mentioned, and he agreed with me, on my own personal observation, that the chromolina odorata is going. It's just, and it's being replaced. You see, imagine new techniques. I was so impressed when he just mentioned it. I'm not a taxonomist. I'm a mycologist. So I was just, it's a personal observation as a botanist. And also, he just told me that the name Econia crassipes has changed. Two things I learned, apart from, and do you know that another thing I learned while we are having this conference, that taxonomists are still adding information on the already stored knowledge. Personally, I didn't know. I thought it had stopped. That is Google that brings this information for us. I didn't know that new knowledge had been added into taxonomy world. Because, those of everybody who started botany, mostly when one white man was teaching us, an Indian was teaching us, Professor Gill, late Professor Gill, he will never pronounce any, taxonomy became a burden until Nigeria started teaching it when we were in Uniben. He was always, I don't know whether as a year one student or year two, you don't even understand much English. Then for you to be hearing volume, you won't even know what you are discussing. So the man made taxonomy to look like, to us, it's another study you will do in heaven, not on earth. Just like another thing that excited me, when people were discussing about AI, for those of us who are Catholics, immediately Pope was warning, he said he's going to delve into the problem of AI. That is his first manifesto. That really touched me. Because no matter what you are hiding, the AI will expose it to you. That's just what touched me. Anything you are hiding. Ajibada was telling me how he asked AI to write the speech he's going to make in the church. To preach for him. AI produced a very nice speech, better than what he knows. I'm only cracking joke with all of us while we are waiting for him. If you are ready, Ajibada, please let me give you my. Thank you. It's a joke. Clap for me for my joke.
Thank you very much, Mr. President and the high table. This is the moment we appreciate the labor of our dedicated editors, the chief editor especially, Professor Namo of the University of Jos. Many of us have sent submissions and they have diligently sorted them and prepared them in these publications. As you know, these publications, they are our legacy and our contributions to knowledge for the benefit not only of ourselves or our country, but the world at large. They are records that posterity will come to meet, acknowledge, and benefit from. So I want to take this moment to thank all our diligent editors, assessors, for all the labor they have put into selecting the papers for publication and going through all the process to produce these copies. Here we have volume 37 of 2014. Volume 37, number 1 of June 2024, sorry. And then volume 37, number 2 of 2024. These volumes included a lot of insightful publications. I don't have time to mention them in particular, but when they get to you, you will realize the amount of effort that has been put into producing them. Every year we face the challenge of the cost of production. And this time around is still the same and even mounting. We supplement the cost you pay for the processing and even publication from other sources. So this is the time to call upon all the patriots that don't want these journals to cease, to please come to our assistance. There was a time Tet Fund was supporting, but that support is no more. And every effort from about 3 years ago, you see us putting into these publications is completely our own. Don't mind that we print Tet Fund on it. It was a deliberate decision, but it is all our effort. So at this time of launching these publications, this is when we call for your support in joining us sacrificially to please sustain these publications. Talking about sacrificially, I hope you know that your assessors are getting virtually nothing in terms of financial return. And the editor is putting in all the time and effort for virtually no personal profit. So at this moment, I'm going to call on Mr. President, to launch these volumes and then open the path for us individually or as an institution to contribute to the launching. At this launching, you can only launch per copy. So there will be no, I pay so amount for 5 copies. No, it's going to be just 1 copy. You can launch for number 1 or number 2. And please realize that even the basic cost of printing is more than 4,000. And let this guide in moving you to launch at a pace or rate that will really help the association. So at this note, I request Mr. President to please launch volume 37 of 2024 copies of the Nigerian Journal of Botany.
Thank you. As the business manager mentioned, that it is really an effort put together which culminates to these volumes. And it is in this note we call on us to come forward and launch this. So I officially launch this volume of the 2. Remember, the cost of production these days is a bit high. I think a copy costed over 5,000. So you cannot buy it, it's less than 5,000. So it's open for general public to launch. So I launch this. I'll take 2 copies at Yes, so I hereby launch the journal. Thank you.
Thank you, Mr. President. Please, a round of applause.
I launched the journal to the public.
You should know that when he decided to keep mum about the amount, is something substantial. Can I invite individuals to please launch these journals?
I think I will take it at 10.
You can raise your hand to signify
Okay, this issue we mentioned should remain as
either on behalf of yourself or your institution. Yeah.
Okay, thank you very much. As I have said, I'm Professor Lon Sani from BIU University, Kano. On behalf of my department, I'm the head of plant science and biotechnology. On behalf of my department, I am picking one of the 2 copies at the cost of 10,000 naira each.
Thank you very much. 20,000 from Professor Sani. Okay. Please, you can pay immediately, either cash or online. The bank account is in the fly pages of the booklet. The bank account is on page 6, Roman 6, page Roman 6. Don't think that your contribution is minimal. Everything from inception in the Botanical Society has always been very highly sacrificial. The founding fathers of the society did not shy from sacrificing.
Please, my HOD just called me, gave me 10,000 naira. So I'm launching for head of the. This man is disturbing me. He didn't want me to talk about my money. Don't you know that to make more is difficult, and to eat it is equally difficult? Eh? So my HOD just called me, so I should just, on behalf of the department, I should just give the Botanical Society 10,000 naira. So thank you. If you will give me one, if you give me 2, I don't mind. 10,000 naira.
That would be just one copy. Yes, please. Please, can you come forward? Please come forward, please. No, I'm coming. I thought I saw your hands up. Did you?
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم الحمد لله رب العالمين والصلاة والسلام على رسول الله صلى الله عليه وسلم رب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقهوا قولي. I, Dr. Umar Tambari, the Provost of Shehu Shagari College of Education, Sokoto, on behalf of my management and the conference participant, on behalf of my college, I want to buy the 2 copies, the July or June 1st publication, and also the December 2nd publication of 2024, 2 copies at the rate of 50,000 naira. On behalf of Shehu Shagari University of Education, I'm also buying the 2 copies at the rate of 20,000 naira. Thank you.
And thank you. On behalf of the Department of Plant Science, Usman Danfodio University, Sokoto, we are buying the 2 copies, the June 2024 Volume 1 and December 2024 Volume 2 at the cost of 40,000. That's 20,000 each.
2 copies, sir. 2 copies, sir.
All right. It has come to my turn, I guess. All right. My name is Dr. Albert Ejikeme from Department of Plant Science and Biotechnology. While we are just seated here, Port Harcourt, University of Port Harcourt. Is there another University of Port Harcourt elsewhere? All right. So we just communicated with our HOD, and he had directed that we should launch 1 copy of each of the volumes that he's presented before us for 10,000. So the 2 copies, 20,000 naira.
My name is Dr. Mrs. C.N. Nebi from University of Nigeria, Nsukka, Department of Plant Science and Biotechnology. We communicated HOD, and he said we should launch the 2 copies with a sum of 20,000 naira. That's 10,000 each for the copy. Thank you.
I am Professor Amatu Aliu from Department of Botany, Ahmadu Bello University, Zaria. On behalf of the Department of Botany, we wish to launch each of the copies with a sum of 15,000 naira, totaling 30,000 naira for the 2 copies. Thank you.
Okay, can I get your number?
My name is Dr. Kolawole Opeyemi Said, the head of Department of Biological Science, Federal University, Kashiri. On behalf of my department, I'll be launching each issue with a sum of 10,000 naira, making 20,000.
My name is Sekinon Sokiola, assist from Obafemi Awolowo Lower University, Ife. We've communicated with our HOD. So on behalf of both department, Obafemi Awolowo University, we are launching each copy for 10,000 naira, make it 20,000.
She has to record it first. Take. We have three. 1, 2, 3. Maru. Maru. 3 persons now. 1, 2, 3. Let me go to her. Hello, madam. Your hands were open.
I am Dr. Gona Mary Timei from Department of Plant Science and Biotechnology, Denis Osadebe University. On behalf of my department, we want to launch the volume B with 10,000 naira.
Thank you. Volume B. Business manager, sir. Volume B. Volume B launched with 10,000 naira. Volume B. Volume B launched with 10,000 naira. Just volume B. Wait. She's already, yes. Volume B, 10,000 naira, yes. I'm coming to you, sir.
My name is Matthew Lawani. I'm an assistant lecturer from the Department of Plant Science and Biotechnology at Bruce Ali University, Ekoma. On behalf of my department, I'm going to launch 2 of the volumes for 10,000 naira. Thank you.
Any other person? Any other university would like to launch?
You're launching it.
Okay.
Good afternoon. I want to, on behalf of the Department of Plant Biology and Biotechnology, University of Benin, we want to take the 2 editions for 20,000 naira.
Would any other volunteer like to launch? Uni Osun. The HOD, University of Osun. Abi Osun State University. Okay. Osun State University. The HOD is here. Is it you? Okay, they have a copy. Osun State University. Eh? How come you have so many copies? Osun State University. The university or the department? The department.
Yes, they already have. They have. See them at the back. They already bought. They launched, I think. We need more launchers, please. Yes. Yes. He said he wants to buy. We are launching now. He wants to launch now. Okay. I'm glad.
He knows how we do
The microphone is yours, sir. Please launch. Actually,
Thank
you've
you.
been pointing other people to launch, so it's your turn now. Thank you, sir.
There is difference between buying and launching. I just want to buy smart. I'm buying 2 copies for the cost price that you just announced. Thank you. The cost price you announced, Professor Ajibade. Mr. President, sir, it's not by size. Okay, I will buy when it is time to sell.
Prof, sir. Prof, sir. Professor Ajibade. On behalf of Department of Plant Biology, University of Ilorin, we are buying 2 at the cost of 10,000 per each, making 20,000.
Thank you, sir.
Thank you, sir.
20,000. Okay. Are there online participants that also want to launch? We will be glad to take them. Online participants.
My HOD should launch you.
I just called my HOD.
Okay.
Gombe State University, Department of Plant Science, and he said he will buy each copy at the sum of 10,000, making 20,000.
Thank you.
Hey, lucky.
It's like we have online launchers. Okay. Yes, we are open to take you online participants that want to launch our journal. Someone is speaking online. We could barely...
Media, please mute.
Okay, media, our online participant, if you want to launch, you can just raise your hand. Please, you can mute them. Thank you. So, if you want to launch online participant, you can raise your hand, and then they will unmute your mic, and then you can launch. Yes, I have a launcher. So, please, mute all, and then when we find a hand raised up for the online participant, we can unmute so that they can launch. Thank you. Yes, sir. Please launch.
Thank you very much. I am Jafar Umar from Federal University, Birnin Kebbi, Department of Biological Sciences. I am buying the 2 copies at the rate of 10,000 naira each, 20,000.
2 copies. Oh, thank you. 2 copies. Can we have more people taking down? Okay, I'm coming, sir. Yes. 2 copies, yes. Thank you. Okay, sir.
Hello, good afternoon, everyone. I'm Prof. Suleiman Ali Anka. On behalf of Head of Biology, Head of Department of Biology, Usman Danford University, Sokoto, we are launching 2 copies at the cost of 10,000 naira each. That is 20,000 naira.
Thank you, thank you. Thank you. More launchers. I saw one hand earlier on. Yes, sir. I'm with you.
Good afternoon. My name is Ajadi Ibrahim from the Department of Plant and Environmental Biology, Kwara State University, Malete. On behalf of my department, I'm launching the 2 copies for 10,000 naira.
Okay. Thank you very much. Please ensure that you document your names with us. Document your name. If possible, do the transfer. Do we have more? Okay.
Kwasu.
Please.
Dr. Ajadi from Kwara State University. Is he here? Okay, he has launched.
Please, if you are paying online and you want to send the evidence, please send it to my WhatsApp number. Please, can you prepare to take the number down? It's 080 59114 705. You can also find the number on the fly leaf of the journals, page Roman 6. The number is there on page Roman 6. So if you want to send the evidence of payment, you can send it to my number. Okay?
Are there more persons who would wish to launch the journal? Are there more? We're interested at the moment for those who want to launch. I'm sure it will be of great interest to you, so you wouldn't leave without buying a copy. I think they are limited. Oh, there are copies. Okay, good. So we have enough copy to cater for you, sir. But let's attend to those who are launching. Yeah, thank you. More? Are there more people who wish to launch the journal? Media, do we have any online launchers of our journal? There is? Okay, yeah, I see one hand raised. Yahoos, yes. Please unmute Yahoos and let him launch. Thank you.
All right, good afternoon.
Good afternoon, Yahoos.
Okay, I am Dr. Yahuza Tenimu from Ahmadu Bello University.
Okay.
On behalf of myself and my family, I'm launching a copy of each for 10,000 naira.
Thank you so much. Thank you, thank you, thank you, sir. Thank you. I believe that has been documented. And I believe that, yes, 20,000. He has launched with 20,000, 10,000 naira for each edition. Okay? Okay, your copy will be given to Professor from ABU. Okay, so you would have your copies. Thank you, Dr. Yahoos. Okay, any more online launchers of our journal? Okay, we'll be wrapping up this session quickly because we need to get into the most interesting session, the plenary session, the breakouts where we will begin to take our presentations. Okay, so we need to really wrap up with this so that we can progress with that. If there are no more individuals that will be launching, I believe, Chairman, sir, we can make some progress and move to the next item on the agenda, which will be please ensure to get a copy, buy a copy before you leave. Between today and tomorrow, we have enough copies that will be. Okay, you want to launch now? Finally.
On behalf of myself, and myself, I'm not launching, but I'm buying the 2 copies for 20,000 naira.
Wow, that's a launch. Thank you. Thank you. Thank you so much. Yeah, we have one last copy. I think our business manager doesn't want to go unless this last copy is launched. So who's going to be the one to launch this last copy? It's not the last copy actually, but just for the launching purpose. We have more, and if you consider launching, we have enough copies. So please, who's going to take this one off the hands of our business manager? Yes, thank you so much, ma'am.
I'm Professor Noye Dema. I'm launching it with 10,000.
Thank you. Thank you so much. We appreciate you. So we've come to the end of this session. There are lots of copies. Please ensure to get one for yourself of both editions before you leave. Today or even tomorrow, they are still available. So everybody should have a copy. But we're moving straight on with the permission of the chairman for the plenary session. He will tell us how we will go. Thank you.
We have 3 venues. We have 3 venues. The virtual session will remain here. We have the College of Medicine session and the postgraduate school session. I will announce the chairman, the respective chairman for the different venues. For this venue, the virtual session, we have Professor K.S. Olorumaye, who will be the chairman. And then as rapporteur, we have, is Mr. Hyacinth around? Obayi Hyacinth? Okay, you'll be the rapporteur for this session. For the postgraduate school session, we have Professor Ramatu Aliyu, and the rapporteur will be Okubo Philippine. Okubo Philippine. Okay, for the Professor Hafsatu Shetu, are you here? Shehu. Professor Hafsatu Shehu, is she here? Yes. Where? Okay, you'll be the chairman for that session. And is it Dr. Hassan or Professor Hassan? Dr. Hassan? Dr. Hassan? Yes, you'll be the rapporteur for that session. So for those 3 venues, we have the chair, respective chairmen, and rapporteurs. And the timekeeper for this session, the timekeeper for this venue will be, is Esther Benjamin here? Esther Benjamin? No? You're here? Okay, you'll be timekeeper for this session. Esther Benjamin. Matthew Lawani. Matthew Lawani. Okay, you'll be the timekeeper for PG School. And the College of Medicine. Onyechi. Is she here? Not here? Okay, you'll be the timekeeper for College of Medicine. So we have all the, respective individuals that we oversee the respective sessions. You might head off to the respective venues. Probably our MC will assist with the location so that they know where to go to. Which one? Ramatu is going to PG school. He's going to College of Medicine. College of Medicine is Professor Hafsatu Shehu.
Where is Professor Hafsatu, please?
You'll be here. It doesn't matter. Just tell them so that somebody replaces you while you present. Are you presenting here? It's on site. Okay, so you can't be here now if you are presenting. Okay. Has been loaded, so you'll be presenting here. You sure? Okay, okay. Okay, go ahead, go ahead. I don't even know. I don't know the venue. I thought they were assistant.
Hello, Deborah. At the school, they just enter the gate.
I understand some of us have problems in locating the venue. I'll just yes, please. Okay, how do you know where you go to? Okay. Give her the
Going to postgraduate school, please you follow me as I go out. Why?
They didn't segregate this thing.
Oh, who are the people? Okay, I wouldn't. Okay, team. Perhaps the team will guide them to know who's going to. Yes, but whenever you find your team, I'm the one to take you to your destination.
Okay,
Thank
for
you.
the PG school.
Yes.
The PG school. Natural and ethnobotany will head there. Natural and ethnobotany, is that what it says? And then for medical school, general botany, we go there. The point is that it was not, they were not segregated. So if your paper is in general botany, you head to College of Medicine. If your paper is related to ethnobotany, you head to the PG school. That's what I have here. Probably we'll need to seek clarification from the LOC chairman. Is he here?
Professor Akoma, your attention is needed, sir.
Okay.
Professor Akoma, your attention is needed, sir. LOC chairman, sir. Okay, he's coming. Yes, he will join us shortly.
Professor, please come and take your seat here.
Hello? Please, I have some copies for authors. I want to read out the names, so if you are the one, you can come and collect. Chukuma Deborah, Federal University, Oyekiti. A.O. Ishola, University of Ibadan. Happiness Uselebe, Ebonyi State University. Apagu Bitrus, Abubakar Tafawa Balewa University. Apagu Bitrus. Catherine Namani, Ebonyi State University. PCI Chijindu, University of Delta. Chijindu, University of Delta. Loretta Ofodile, Yaba College of Tech. Ben Uwalaka, David Umahi Federal University of Health and Sciences, Ebonyi. CN Dada, Obafemi Awolowo University. Sanusi Kabiru.
So are we breaking for lunch?
Sanusi Kabiru, Nigerian Army University. Ben Uwalaka, David Umahi University. GK Olawepo, University of Ilorin. BS Aliu, Federal University of Tech, Babura. OT Oladele, Obafemi Awolowo University.
Professor Akoma, sir, LOC chairman, your attention is needed upstage. Thank you.
VO Ikani, Modibo Adama University. Loretta Ofodile, Yaba College of Tech. VO Choir.
All right, please, my dear BOSON conference participants, there is a modification in the program. For some logistical reasons, we want to take all virtual presentations while we're here today, this evening. The much we can take between now and the next 1 hour, and then we disperse. While tomorrow morning, we start with the breakout sessions in the various venues, while this location is dedicated to the remaining virtual presentations. So we will be here, and then the virtual participants that have been queued up for presentation, we'll take them while we become the audience for them today, 100%. Tomorrow morning, we'll break out to natural products and ethnobotany section, ecotoxicology and general physiology, and then other sections. We have 4 breakout sessions, and then this venue will also be dedicated for virtual presentations. Thank you very much. So let's start with the virtual presentations and no physical presentation for this evening. Thank you, sir.
Where your guy? Ma? Where your guy? Yes, sir. Call am for me.
Hello? Is there any virtual participants online that want to make presentations now?
Yes, we are online. We are waiting for you.
Please, as part of the condition for presenting, we want to know if each participant has made their payments together with the membership payments, the dates of the payments, and you will be asked to present immediately.
All right.
Please, your name?
Okay, I'm Benga Komalafe.
Benga Komalafe, I think his name is Pato. Benga Komalafe, I think his name is Pato. The date of payment of the membership fee.
I paid on 9th of June.
9th of June. Yes. Who is the cherry?
David.
So, Dr. Komolafe, are you ready with the slides? Please, the media people, please, can you bring up Dr. Komolafe for his presentation? Thank you. How many minutes for the presentation? 10 is not too much. 7 presentation and 3 for. Dr. Komolafe, you have 7 minutes for presentation, and there will be 3 minutes for question and answer. You can start now.
Okay, thank you so much. Greetings to the chairperson of this session and all other protocols duly observed. I'm Benga Komalafe from Federal University of Lafayette, and I'll be presenting on land use change and vegetation health assessment of a dry tropical montane forest reserve in Nigeria using satellite remote sensing. And this is the outline of my presentation. Of course, we know that population growth is one of those factors that have intensified land exploitation globally. And land use and land cover changes are one of the major contributors to global environmental risk. One of the things that, one of the latest technologies for assessing land use and land cover is the use of remote sensing, and particularly through the production of thematic maps, whereby we can assess the rate of land use change, and then, of course, vegetation change globally. And Nigerian protected forests, particularly the one I'm looking at today, is Ingeli Inyake Forest Reserve, which is actually located in Taraba State. These protected forests, they have actually faced numerous threats that endanger their ecological integrity and sustainability. And of course, there is not much studies done on evaluating the extent and the drivers of land use and vegetation change in some of these protected forests, particularly the Nge Inyake Forest Reserve in Nigeria. So the aim of my study actually was to look at the land use and vegetation change of this forest in the last 30 years using remote sensing technology. And in the course of the research, I looked at the land use and land cover change of the catchment area where the forest is located, which is the Sardauna local government in Taraba State. And of course, I also looked at the land use and land cover change of the forest reserve itself, and then I also evaluated the vegetation dynamics of these forests over the past 30 years, from 1993 to 2023. And this is the methodology of the research. We obtained satellite data, and this satellite data actually were obtained from the USGS website, Landsat particularly, and I used two different types of Landsat satellites. The first one is Landsat 4 and 5, and then the second one is Landsat 8. And this satellite data have actually been used by so many researchers, for the classification of land use and land cover in so many parts of the world. So they've been found to be very useful and very, very accurate. So this is a map of the study area as well, which is the Nge Inyake Forest, and then the Sardauna local government, and then the Taraba State itself. So the study, like I said, after collection of the satellite data, we did some pre-processing in QGIS, and then, of course, which involved quality checking and so many other GIS techniques. And after that, we proceeded in using these satellite images to classify the land use and land cover of the Sardauna local government and then the forest reserve itself. And for the forest reserve, we focus mainly on three different types of vegetation: the dense forest, the moderate forest, and then, of course, the open land land use. And then for the catchment area itself, we focus on the dense forest, the moderate forest, the open land, the built-up areas, and then the wetlands. So these were classified using what we call the maximum likelihood algorithm in QGIS. And after classifying the land use and land cover, we also went ahead to to analyze the vegetation change over these past 30 years. And we use some of the vegetation indices, which are the NDVI, which is also called the Normalized Difference Vegetation Index, the EVI, Enhanced Vegetation Index, the NDWI, Normalized Difference Water Index, and then the Soil Adjusted Vegetation Index. And then lastly, we also did the chlorophyll, the green chlorophyll index rather. And so these were the vegetation indices that were used to determine the LD status of the forest. So the result, we discovered that in the last 30 years, the local government area, which is the catchment area, actually reduced in the dense forest reduced to about 6.24%, okay? And then the open land increased. We discovered that the open land increased from what it used to be, from 2.97% to 4.49% in 2023. And we also discovered that this increase in the open land of the catchment area actually affected the dense forest of the dense forest vegetation of the reserved forest itself, which means that because of the expansion of probably the catchment area, because of the increase in population or something, then there have been encroachment into the forest, which made the dense forest to have reduced. So for the forest itself, this is the land use and land cover of the forest itself. And of course, what we observed in the general, the entire catchment area, we also observed the same trend in the forest, whereby the dense forest reduced to from 63.4% to 12.68% of the land area. And then the open land also increased. And these were the things that we observed. And we also discovered that most of this loss in the dense forest vegetation of this reserve forest was due to farming activities and some other activities that have degraded the forest. And so to round up, the vegetation indices also showed decrease in the chlorophyll content of the forest and then the greenness of the forest. And this is what we observed, that this forest has been degraded, and there is need for stakeholders to come together to formulate strategies of protecting this forest and also conserving the species that are found in the forest. Thank you very much.
Thank you very much, Dr. Akomolafe. On your presentation on relating land use change to vegetation change in Doma Forest Reserve. Thank you very much. Let's give him a round of applause before we treat him to question. Any question from the audience, please? Question. Any question? Well, let me ask this question. How did the degradation of the land or land use account for the depreciation in chlorophyll content of the plants?
Okay. We believe that, because of the analysis that we did, we actually did some analysis, and we discovered that the changes, because of the loss of the greenness of the forest, the chlorophyll content also decreased, which is actually logical, because the greenness can be related to the chlorophyll content of the vegetation. So if the vegetation has lost a large number of, a large area of the forest, definitely the vegetation would have also decreased in chlorophyll content.
Well,
so this is actually what we
Thank you very much. Maybe in a subsequent work on that, try to include an anatomist. Maybe the process of the land degradation through excavation. Most of the chlorophyll opening, most of the stomata opening must have been blocked by the dust of the soil excavated. And if such happened, carbon dioxide will not be able to enter for photosynthetic activities. You can still investigate that too. So,
thank you, Prof.
Thank you very much, Dr. Komalafe. I know for sure that plant response to changes in environment, whether biotic or abiotic, are species specific. Will you say this was considered in your study, or it was just a general assessment with respect to chlorophyll content? Because we have done a number of studies with respect to responses of plants with either land use change, mining activities, even forest gap. And we know that these responses are species specific. Can you confirm that, please?
Okay, thank you. Yes, you're correct. But our study was actually based on a general, we didn't focus on the specific status of the species, but just a general forest assessment, looking at the greenness of the forest generally. Thank you.
Thank you very much. In the absence of any further question, thank you very much for the presentation. Let's appreciate him for trying to safeguard our plant from becoming chlorotic as a result of abnormal land use. Thank you very much. The next presenter, please.
Good afternoon.
Please, the presenter, please. What's your name?
Yeah. My name is Oyade David, Dr. Oyade David, Federal University of Ekiti.
Please,
so,
what's the date that you make your payments, your membership payments?
This morning.
This morning? Oyade David.
David.
Dr. Oyade David. Yes, thank you. You have 7 minutes for the presentation. Thank you.
So, good afternoon, everyone. And my name is Oyade David from Federal University of Ekiti. And I'm here to present my research study titled Silicon Nanoparticle Promotes Tomato Root Response to Flood Tolerance. So, flooding crisis in Nigeria, we are all familiar with this incident that happens in year 2022, where we have a climatic disaster where there is damage of properties, farmlands, and lives was lost. And we can see from the image showing the submergence, complete submergence of all the properties. And this is the likelihood damage flooding can do to the environment. And then what do we anticipate in the next 2050? In the next 2050, it is expected that there will be an increase in amount of precipitation that will be received, especially in Africa, from 15% to 54% compared to the usual precipitation that we have been receiving, which indicate that there might be a probability of increase in more associated risk related to flooding. And also, it has also been said, it has also been reported that flooding frequency, intensity, and duration might also increase in the 2050. And so, what happens to plants under water? Simple. What happens to plants is that they find it difficult to breathe, especially the root part of the plants. And so this is what we call oxygen deficiency, known as hypoxia. When it is completely absent, it is called anoxia. And in this process, plants put in so many metabolic activities in order for them to survive. And one of them is to convert their aerobic respiration into anaerobic respiration in order to limit the energy metabolism during this process. And for them to be able to cope in this process, they have two ways, majorly two ways, which is what we call an escape, or they go into a quiescence. Now, escape is when a plant uses some anatomical parts of its plant in order for them to adapt to this condition. And one of them is epinastic, which is the downward movement of leaf, which is controlled by hormone ethylene. They can develop erythema, which is a hair space for them to be able to breathe better and develop adventitious roots in order for them to survive during this condition. And under this process, they can also change, they can induce some hypoxia gene, which can help them to survive. That is a response to this condition. And also there can be an accumulation of antioxidants enzymes during this process. So how may silicon aid flooding? Silicon is a non-essential element. However, it has been reported that they can help to plants tolerate green abiotic stress such as drought stress, salinity, with an evidence of them increasing antioxidant activities, which helps to scavenge reactive oxygen species during this stress. And also, one of the enzymes that is involved in production of lignin, which is peroxidase, is also accumulated, knowing that during the stress, plants can, or silicon can help plants to accumulate more lignin in order to reduce radial oxygen loss during this process. Though silicon is not essential, it is not really necessary for the plant growth, but it is important during abiotic stress. And so we decided to use nanoparticles because of the size and the absorption surface area. And we synthesize our silicon nanoparticles from rhizomes using the process of calcination. And so this is how we get the silicon nanoparticles. And this is the process of its uptake in the soil. When it gets into the soil, it's get accumulated at the exodermis part of the plant using an influx transporter. And when it gets to the endodermis, it's also changed to an exflux transporter, which is low silicon 2, which helps to move the silicon from the endodermis into the shoot part of the plant. But tomato lack the exflux transport, so meaning that the silicon can only get deposited at the endodermis without moving into the shoot part of the tomato. And that is what informs us in the use of silicon, in the use of the nanoparticles, if it could probably help us to move the silicon up to the shoot part of the plant. And so the aim of the studies is for us to unravel the effect of silicon nanoparticles on a flooded tomato roots architectural. So we try to characterize our silicon nanoparticles using some instruments. We use some SEM and we use EDS to characterize. This shows that we have actually synthesized silicon nanoparticles less than 100 nanometers, pseudo-spherical shape, and we are able to observe that we have above 50% silicon present in what we have synthesized. Aside silicon, it's only carbon and copper and oxygen that is present in what we have synthesized, which shows that it is eco. Okay. Oh. And so we carry out some experiments and realize that silicon helps to induce the growth and adventitious growth of the plants, and also it helps in survival rates of the plants. And we did the water logging again, and we saw the same results. This is the root architectural that we did using, we did the rhizobox experiments, and we tried to do the root analysis. So from the root analysis, we observed that during the flooding, silicon helps to increase the main root length, the lateral root length, the lateral root density. We did the distribution, the root distribution, and realized that silicon helps in the development of lateral root density at the upper part of the plant. And also realized that there is at the post-recovery, we observed that the root of the one treated with silicon was a little bit deeper compared to the one that was treated with flooding without silicon, which was shallow. And then we did some flavonoids, and we carried out some exodermis tests using lignin. We stain our plants, and we try to use some exodermis, the signals, using confocal microscope. And we observed that there was the exodermis part of the tomato plant was more magnified and the intensity of the signal was higher at the upper part of the wood and the lower part of the wood, and it is the thickness of the exodermis was even higher in the lower part, which is so. This is the sublime deposition of the silicon nanoparticle, which also shows signal. So my summary says silicon nanoparticle increased adventitious roots. Roots growth and development was not significant. Flavonoids was also induced or accumulated with the help of silica. Lignified exodermis was also observed and accumulation of sublime. In conclusion, aside the formation of adventitious roots, we realized that lignified exodermis and at the upper lateral roots can be a trait for plants to cope with waterlogging stress. And also we realize that sublime and accumulation of flavonoids can also be of a help, which we all know that is an apoplastic way. It's trying to create an apoplastic barrier for them to be able to survive the
Thank you
stress.
very much for the presentation.
Thank you.
Thank you very
so
much. A round of
Thank you.
applause for Dr. Oyade David. Any question from the audience, please?
Hello, thank you for your beautiful presentation. I'm Professor Ajibade. You did mention that silicon is not really essential for the growth. So how
It's not a
practicable,
limit factor.
how
Okay.
practicable then is this innovation in our situation where flooding is unpredictable?
Okay. Thank you so much. Yes, according to the literature, we all know that silicon is one of the abundant elements in the soil. And so then it is not every plant that can transport this silicon. We see that rice can be able to move this silicon very well, but however, tomato plants have this difficulty. So what's intent of our research is for us to, okay, is there a way we can synthesize something that is eco-friendly and something that can help plants to respond better during flooding and able to survive and able to recover and regenerate over a period of time? So this is what points us to the use of nanoparticles, being able to deliver silicon easily to the plants and it's absorbing surface area, and that was the reason why it prompted us to use nanoparticle instead of even using the box sample of silicon. I don't know whether I've answered the question.
Thank you very much.
Thank you, sir.
Yeah, many thanks for your presentation. I'm Dr. Dan Dosibe from UNN. So I'm wondering if silicon nanoparticles you synthesized are transported similarly with the normal silicon in the soil. So my question basically is, the nanoparticles you synthesized, are they transported with LSI1 and LSI2, LSI6 using similar transport mechanisms like usual silicon in the soil?
Yes, for tomato particularly, they are based on the literature. They lack low silicon 2, which moves silicon from the endodermis to the shoot part of the plant. But then it can get deposited at the roots, which the roots can induce some signals. So the silicon, because we did some molecular work, we realized that it induced some signals that can help plant to survive. So we have actually not established whether the silicon actually moved to the shoot part of the plant. But then we've established that the silicon is deposited at the endodermis and at the exodermis of the tomato plant. But we have not actually, based on what we have done, we have not actually established whether it has moved to the shoot part of the plant or not. But according to the literature, the tomato plants lack transporter to the shoot part of the plant. So then we want to see if nanoparticles can help us to move this silicon to the shoot part of the, but for now we have not yet established that, but we are rightly on the root part of the plant. So with the work we have seen at the root part of the plant, we observe that it is able to induce ethylene. And that's why we're able to see more of adventitious roots produced, because adventitious roots and erythema are response of ethylene. And so that's why we can able to see more of adventitious root developed when silicon was applied into the root part of the plant. But then we have not yet confirmed the transporter to the shoot part of the plant. And that's why we made it at the root level for now.
Are you okay? He said the question.
What I'm saying, because LSI1, LSI2 were identified from rice. So, so what is the expression of these genes in tomato? And my question is, can tomato take up silicon nanoparticles just the same way they take up the ubiquitous silicon in the soil?
Yes, they can, up to the root level, according to the literature. They can take up silicon up to the endodermis part of the roots of the tomato plants, unlike rice, but then to the shoot part of plants, we are not sure.
I'm Tanko James Agwe, even though I came late, but I caught across a slide that says flavonoids buffers ROS. So I wanted to know, are there other secondary metabolites that can buffer ROS based on the research you've conducted, or specifically you focus on only flavonoid? Thank you very kindly.
Okay, we actually, aside the flavonoids, we actually look at anthocyanin, but then we have to look at the consistency. When we look at the anthocyanin, it was not that consistent in our research because what we have presented, it has been done like 3 times, 4 times before we make our conclusion. So, but then anthocyanin keep on increasing and decreasing, so we can't make a conclusion that it's actually due for anthocyanin, but flavonoids was consistent all throughout the experiments, even while we are doing the flooding and while we are doing the water logging, and even because we carry out 3 experiments, water logging, flooding, and in a rhizobox. So, and we realize that flavonoid gives us a consistent accumulation of flavonoids. And so that's why we were able to say, okay, flavonoids can be induced during flooding, and it could be responsible. According to literature, we have so many other antioxidants that can help to scavenge reactive oxygen species. Aside flavonoids, we have phenolics. They are also there. They can help to scavenge reactive oxygen species. Then we have glutathione. We have so many catalysts that can be there to scavenge reactive oxygen species. But for this purpose of this experiment, we
Thank you
only
very
work
much.
with flavonoids and
Thank you very much. I want to ask this. Did you take cognitive of the oxygen use efficiency by this your nanoparticle? Because the more water we have in the soil, oxygen will be totally displaced. And maybe the reason for the adventitious root development may be as a result of the respiratory activities, breaking down food for growth and meristematic activities, cell diffusion activities that are responsible for that. So as you progress in the experiment or the research, try and see whether oxygen is more available as a result of this nanoparticle for respiratory activities. Since flooding will expel almost 100% of oxygen in the soil. So thank you very much for the beautiful presentation. Thank you. A round of applause for him.
Thank you very much.
You can step down. Thank you for the beautiful presentation once again.
Thank you. The next presenter is Mohammed Jamila. You have 7 minutes for presentation and 3 minutes for question and answer. After Mohammed Jamila, the next presenter will be Abigail Damfami. After Mohammed Jamila. Then after that, we have Chima Obiefule. Thank you.
We are not hearing you.
afternoon, sir.
Good afternoon. We are hearing you.
Yes, I'm trying to share my screen, sir.
Your time is already running.
Okay, sir.
Are you Mohammed Jemila Abimbola?
Yes, sir.
Okay. Go on. We
Yes,
are
sir.
listening to you.
Okay, sir. So, I'm presenting on the history, uses, and benefits of tobacco, nicotina tobacco.
Your screen is causing a kind of reflection. Do you have any bulb on there or window?
There's no bulb, sir.
Continue. We are
So, I'm going to start. Nicotina tabacum is the Latin name for tobacco plant. Originally, Nicotina tabacum came from the North and South America, but it is now grown almost all over the world. The plant is easy to grow, used as an ornamental plant and for making tobacco. It's tobacco is one of the biggest public health threats the world has ever faced, killing more than 8 million people a year, including around 1.2 million deaths from exposure to secondhand smoke. Tobacco plant is perennial, erect granular pubescent herbs and found only in cultivation. Nicotina tabacum is basically a native of America, but it is now commercially cultivated throughout the world. The other variety of tobacco plants are cultivated as ornamental plants or grow as weed. The written history of tobacco begins in the year 1492 when Christopher Columbus discovered American Indian treating their ills with the leaves of herbs, which he had never seen before. The genus derived its name from J. Nicot, a Portuguese who produced the tobacco plant into France. Tobacco was introduced to Spain and Portugal in the mid-16th century. It spread to the other European countries and then to Asia and Africa, where its use became general in the 17th century. It was considered a decorative plant at first, then a pinaceous before it becomes a common snuff and tobacco plant. Native American shams developed tobacco use for religious rites. The antimicrobial activity. So, ethanolic extract of Nicotina tabacum used as medicinal plant extract in treatment of tuberculosis. The nicotine-like acetylcholine is known as number of sensory receptors. Traditional use of tobacco. Tobacco has been used as antispasmodic, irritant, diuretic, emetic, sedative in homeopathy. Tobacco has a long history of use by medical herbalists as a relaxant, though since it is highly addictive drug, it is employed internally and externally at present. Where tobacco leaves are applied externally in the treatment of rheumatism, swelling, skin disease, and stinks. A homeopathic remedy made from the dried leaf is used in the treatment of nausea and motion sickness. Some other activities report for Nicotina tabacum are analgesic activities, antimicrobial, antibacterial activities, anticonvulsant activities, antifungal activities, antioxidant activities, and anti-stress effects. Nicotina for treatment of rosacea disease, depression, anxiety, attention deficit, hyperactivity disorder, pain, and obesity. The
You
country
have
has
2 more
the country
minutes.
has that
2
have
more
history
minutes.
of. Sir?
You have 2 more minutes.
Okay, sir. All right, sir. So some of the countries that has history use of the plant, including China, the traditional use of tobacco as an agent to detoxify nicotine. Then in India, for muscle relaxation associated with joint dislocation. Then in Nigeria, hot water extract of the fresh leaf is taken orally as a sedative. So all parts of the plant containing nicotine, which can be extracted and used as an insecticide. The juice of the leaf can be rubbed on the body as an insect repellent. Nicotiana tabacum is the main species that is used to make cigarette, cigar, and other smokable tobacco preparation. A drying oil is obtained from the seed. Short-term effect tobacco products, including cigarettes, cigar, and snuff, etc. Then the short-term effects of chewing or smoking tobacco, including excess saliva production and drooling, bad breath, difficulty in chewing, stain and sensitive teeth or tooth decay, damage to respiratory system, addiction to nicotine. Then the long-term effects include cardiac complication, lungs disorder, anti-fertility, and cancer. Other health complications include maternal smoking. Smoking during pregnancy can lead to miscarriage, stillbirth, sudden infant death syndrome. So smoking can cause death disturbance, which in turn exacerbate other diseases. Then conclusion, tobacco can cause addiction and dependence, but it has many practical follows traditional medicinal use. If it is used in positive way, it had power to heal and protect. But if misused, it had the power to harm. It possesses various pharmacological activity. The ethnobotany use include the use of the decoction of the leaf as antispasmodic, diuretic, emetic, sedative, and in rheumatism swelling, antimicrobial activities, and the antibacterial activities. The plant Nicotiana tabacum has great activities on peripheral nervous system, and central nervous system. That comes to the end of my.
Thank you very much for the beautiful caution through your presentation. A round of applause for the presenter. Thank you very much. Questions? Yes, sir.
I just add your comment. You said Nicotiana tabacum is an ornamental plant. I would rather prefer you to say that it's an household plant. That one is better.
All right, sir. I'll note that, sir. Thank you, sir.
Any other question or comment?
Thank you for your beautiful presentation. I'm Professor Ajibade. I'm just wondering what motivated you to doing this review on tobacco and not on any other plant?
Yes, sir, because tobacco is one of the most smoked, like it's made with tobacco is one that they use for to make cigar and it is like that. So they is usually common in the society.
Are you done? Okay, thank you for your presentation. My little question is the acetylcholine content of tobacco, which you mentioned just now, is it of benefit or not of benefit? What will you say about it?
Sir, it's actually a beneficial acetylcholine itself is a beneficial neurotransmitter in the human body. So it plays a crucial role in muscle movement, memories, and many functions of the nervous system.
So all of them are benefits. The acetylcholine is
Yes.
all beneficial.
Yes, sir.
And you said is receptor what? What's the function?
Neurotransmitter.
Transmitter.
Yes, neurotransmitter in the human body.
Neurotransmitter.
Yes, sir. Yes, sir.
Okay. But I don't think so. Thank you.
Yes, sir.
Thank you very much, Ma, for the beautiful presentation.
Yes, sir.
A round of applause for her once again. She's a mother warning our youth not to smoke this. Thank you very much.
Yes, sir. Thank you, sir.
Your time is, you can step down. Thank you very, very much.
All right, thank you so much, Mohammed Jamila, for your presentation. I'll call on the next person, Abigail Damfami. D-A. Looks like Abigail is not ready. So, Chima Obifule.
Chima Obifule.
Yes. Chima Obifule.
Yes. Good evening, sir. My presentation is on ethnobotanical survey of some medicinal plants used in traditional healthcare in Alkaleri, Toro, and Garazo local government areas of Bauchi State.
Chima, share your slide.
Yeah, I'm doing it. I'm doing it while I'm talking, sir. So the ethnobotanical survey are crucial for documenting traditional uses of medicinal plants and identifying active compounds. This is Nagavi and 2021. Traditional medicine remains integral to health care in many African communities, particularly in regions with limited access to modern facilities. Maho Modali, 2023. Many factors strengthen traditional medicinal knowledge and plant biodiversity. Conservation strategies are needed to protect medicinal plants. Mesby and Yarara, 2022. Environmental degradation and deforestation pose risk to indigenous knowledge of medicinal plants. Mesby and Yarara, 2022. Bauchi State holds rich but important documented medicinal plants at Adamu Eta 2025.
Good
Okay. Now, despite investigated medicinal plants using traditional local government areas of Nigeria between April and November 2024, despite availability of modern medicine, many rural communities still rely heavily on traditional remedies. The study designed ethnobotanical survey focused on traditional medicine custodians in Bauchi State. It explored how medicinal plants are used to treat diseases across three local government areas: Toro, Northern Zone on latitude 10.4465 and longitude 9.2206, Garazo Central Zone on latitude 10.9992 and longitude 100 and academic southern zone, latitude 10.2597. The study was carried out in selected communities, including the Chifu LGS, then the Hausa, Fulani, and the dominant ethnic, followed by other tribes like Badawa, Kanuri, Kare, Jarawa, Sayawa. These communities are located in largely agrarian, the long-standing presence of herbal medicine practitioners. The map of the area was created using UGIS 3.22.14, while the Wiser, your GIS 2023. Data collection. Baba informed consent was obtained before interviews were carried out. The structured questionnaires, guided interviews, consulted the Hausa and other local languages with the help from interpreters. The data collection method adapted Martin's 1395 approach using purposive sampling to select 10 communities known for their traditional medicine practices. Rapid rural appraisal with village leaders helped to identify these communities. Globally sampling in Taluwe 2010 was used to reach 150 informants, including herbalists, vendors, and collectors. Data collected will cover plant species using methods and availability. Field work with informants allow for plant collection and virtual specimen preparation. Plant identification and virtual specimen. Initial plant identification were done using guides at the 1999, while full identification and validation was carried out at the Abaka Federal University of Maryland. Scientific names were confirmed using the topical pattern of www.topical.org. Examination of the daily level. The daily level were calculated, the reliability of fitting particular element. IP over IU and content, where IP is the number of informants citing a specific element, and IU is the number citing it for any use. Friedman et al. 1986. Data analysis is fitting statistics using frequencies and percentages were used to summarize data using appropriate software. Now, the results were as follows. If you follow the, according to the gender, the bar charts, when it was subjected to statistical analysis, figure 1 there shows the distribution of respondents by gender. The result shows that the majority of the respondents are male in all the local governments study area, with proportion of females respondents. Now, figure 2 shows the distribution of respondents according to age. The result shows that majority of the respondents are within the age range of 31 to 40, followed by 21 to 30, followed by 11 to 20, followed by 40 to 50, and 51 to 60 and above. Now, within the age of across the local government, overall, these responses do not follow a uniform distribution. Now, figure 3 is the distribution of the responses according to occupation across the 3 local governments. The results of the analysis above shows that the majority of the respondents are Okada riders, followed by herbalists, and so on. In conclusion, based on the results above, responses do not follow a uniform distribution.
A minute more for you.
Figure 4, distribution of the respondents by educational level.
1 more minute. Go to your conclusion. We are not hearing you again.
Is network? Okay, I'm on figure 4. Are you hearing me now, sir?
Conclude.
Okay, sir. Now, in conclusion, or if we can discuss the results, the dominance of Of the male informants, 78% align with earlier observations by Erinoso and Awori in 2012, who reported traditional medicine practitioners and herbalists to be mainly males. This is to say that males are more often the public custodians of indigenous knowledge in northern Nigeria. However, the participation of women, 22%, also highlights the under-recognized but critical role they play in traditional healing, particularly within household. The age distribution of respondents with the highest representation, 31 to 40 age group, suggests that traditional knowledge is actively being practiced by a younger generation. This contradicts fears of knowledge erosion expressed in earlier studies such as Adefia and Obo 2012, which emphasized the loss of oral tradition due to globalization and urban migration. The primary modes of knowledge acquisition, tutoring 40% and family transmission at 7%, confirm the continued reliance on oral and community-based learning system in line with Anoko Bongo 1992. Occupational diversity among informants, including herbalists, vendors, farmers, and collectors, indicates that the practice of traditional medicine transcends professional boundaries. Similarly, findings have been documented by Ukwane Kwaja et al. 2024, reinforcing the communal nature of the
time is off, please. The time is off.
Thank you, sir.
Conclude in a sentence.
Okay. Finally, the study reveals a vibrant and deeply embedded tradition of medicinal practice used in the rural communities of Bauchi State. Despite the availability of modern healthcare facilities, local populations continue to rely heavily on traditional medicines, which are perceived as accessible, affordable, and culturally congruent.
Thank you very much for this beautiful presentation to bring botanical use as alternative medicine into the doorstep of everybody. A round of applause for the presenter. A round of applause. Madam. Any question, please? Any question, please?
Yes, I have a question, sir, and I have a comment. Hello, sir. Am I audible?
Okay.
Yes, I'm hearing you, sir.
Yes.
Yes, I'm Dr. Adeyemi Sherif Babasunde from University of Ilorin.
You are welcome, SB.
Thank you, sir. Yeah, thank you for the presentation. It's quite revealing. However, I just have a comment about the methodology. Sometimes, if we are doing this kind of work, it's better we use up-to-date tools to analyze our data. We have what we call ethnobotany R, which will give you all the index that will make sure that you are, because when you were discussing, you said you were trying to go against a study that said there is knowledge erosion. Sometimes with the kind of analysis you did, you cannot conclude that there's no knowledge erosion because you don't know the quality of knowledge being shared by the younger generation. If the knowledge is quality to what is being shared by the older generation. So when you use all this R, ethnobotany R is a package in R environment, you will see several indices or several ethnobotanical indices that will tell you quantitatively what you should be expecting in your ethnobotanical. Not that it will calculate several indices. So for you to come to the conclusion that, okay, the younger generation have quality knowledge about ethnobotany. Really, the study is a worthwhile one, and I give kudos to you. But sometimes after you go to the field, when you come back to the lab, just make sure you use up-to-date tool to analyze your data. Thank you very much.
Thank you very much, Dr. Adeyemi from South Africa. Thank you. Any other question? A round of applause for the presenter, please. Thank you very much.
All right. The next presenter is Mustafa Yusuf.
Dr. Mustafa Yusuf, we are waiting for you. Are you online with us?
Yes, but hello, can you hear me?
Yes.
It's Mustafa Yahuza, not Mustafa Yusuf, sir.
Go ahead.
All right, sir. Let me quickly share my screen. Mr. Chairman and all other protocols observed. My name is Mustafa Yahuza Suleiman. I'm here to present the topic relating land use change to vegetation change in Duma Forest Reserve. All right, sir. Now, land use changes basically are changes that occur due to certain requirements or certain services that are expected from the general population. But the basic information about land use change is that it is the material expression of the environment and human dynamics in which land is like the mediating factor in this association. And land use changes has a lot of consequences in which it can result in changes in industrial structure. It can either cause employment and unemployment. It can cause poverty and population change. It also affects the quality of life and issues such as health and amenities. Now, I use remote sensing to assess the LULC changes in Duma Forest Reserve because it is accurate and cost effective, and you can use it in every biodiversity related characteristics like monitoring of species distribution, pressures from unsustainable management, pollution change and climate change, ecosystem services, and even conservation status of protected areas. Now, my study site is Duma local government in Nasarawa State. It has a population of up to between 400 to 700,000 individuals. The main activities in Duma are mining and agriculture. Now, my methods and materials, species data acquisition was obtained from the United States Geological Survey. Georeferencing techniques such as cloud removals and image clarification were used using QGIS GIS softwares. Image classification and classification accuracy assessment were also carried out for this study. Now, in the classification, Duma Town was classified into 5 different classes. We have the dense forest, which has a tree canopy of 70% and above. Moderate forest has a tree canopy of between 40 and 70%. Build-up areas are your urban areas and your rural areas and farmhouses. Wetlands are natural and artificial ponds or lake. Open land are areas with low or no vegetation. Now, for my result, this is the satellite images and based on the classification I mentioned of Duma local government area. Now, the dark green colored is your dense forest. The lighter green represents your moderate forest. Open land is the white shades that you see, and then build-up areas are the red colors in the diagram. And I failed to mention that this is assessment in a span of 30 years between 1991 and 2023. So this is the representation of these 5 classes I mentioned in 1991. Now, this is the representation of the same Duma local government in 2023. Immediately, you can see significant changes in the diagram where the red, the build-up areas has an exponential increase. Open land area too has also greatly increased. And if you can observe, there is even no dark green colored area which represent the dense forest. This is further clarified in this table where the change in area were calculated. If you can see this highlighted red portion represent the change in dense forest. We have 100% change in dense forest. It means in 2023, there were no tree canopies that had 70 and above canopy density. Moderate forests also had reduction of 14%. Open land had an increase of 600%. So this shows you how severely degraded the forest reserve is facing. Now, This also represents an accuracy assessment and kappa coefficient. This basically, kappa coefficient basically tells you if your classification of this LUSC class is accurate, and kappa coefficient ranges from 0 minus 1 to 1. So anything from 0 to 1 is shows you accuracy, and anything less than 0 to minus 1 shows low accuracy. So you can see from our classification in 1991, a kappa coefficient was between 68 and 98%. The overall accuracy here, which shows 86%, shows you if your reference site has been properly mapped. So anything below 40% usually in our classification and our mapping showed 87%, which shows high accuracy, and this represents the accuracy for the year 2023. You can see that in dense forest, there's no kappa coefficient because the dense forest is not really in existence now. We had an overall accuracy of 73%, so this shows that the study and the interpretations were accurate. Now, in conclusion, it's evident that this study has been able to use remote sensing to assess the land use and land use change. And from this study, we can conclude that the increase in build-up area ultimately is a result of increase in human population, which is associated with the demand for urbanization. Now, the rapid increase also in population will also lead to the rapid conversion of land for agricultural use. Because I said Doma, one of the major economic activities in Doma is agriculture and mining. So the increase in population ultimately leads to increase in conversion of land for agricultural use. This eventually leads to implications such as increase in greenhouses, loss of vegetation cover, and loss of biodiversity. So stakeholders and management involved have to have aggressive reforestation project initiated if we truly want to save the biodiversity and vegetation cover of Doma Forest Reserve. Thank you for listening.
Thank you very much for the presentation. A round of applause.
Thank you, sir.
A round of applause for the presenter, please. Thank you very much. Any question or comment to the presenter? Questions or comment?
Thank you very much for the presentation on the land use, land cover change. I have a comment and a question. The first comment is that if you look at your title slide, the word vegetation there is not correct. So you may need to check your slide properly next time before.
Yes, sir. Thank you for the observation, sir.
Like this. So I want to ask, how were you able to ensure that you minimize error comparing the ground information to that you obtained by GIS survey?
Okay, that is the table I was presenting, the accuracy assessment. This is just for assessment of accuracy of the data that was provided. And the kappa coefficient, like I mentioned, is what you use to know if your classification, that's the dense forest, moderate forest, and open land, were classified accurately. And you need a point of, it is between -1 and 1. So anything above 0 shows high accuracy in classification. And if you can see in 1991, our classification ranged from 0.55 to 0.99, which shows accuracy in that classification. And also in 2023, we also have a high kappa coefficient, so it shows accuracy in our classification. Now, the overall accuracy represents the site that you reference, whether you have mapped it correctly, and it represents the ground truth data. It is usually estimated with the GPS coordinates of the ground truth data of the site that you are assessing. And anything below 70% will show you that the accuracy or the interpretation of your image from the satellite was wrong. But anything above 40% shows you a high accuracy assessment. And we have 73% in 2023, and we have 86% in 1991. So this shows that our data was accurate.
All right. Hello. Please, I want to ask, how did you compare the GIS coordinates, the GPS coordinates? How did you compare it with the real ground data that you obtained?
Come again, sir?
Hello, please, some people are making noise. Please mute those people that are making noise at the background. You know, what was the sensitivity of your GPS that you used?
Sir?
What was the sensitivity of the GPS that you used, the coordinate point?
The coordinate point?
Yes, to the nearest meter. You know, if you are taking coordinate points, you have to ensure that you minimize error point-on-point basis to at least 2 meters, so that you can be able to effectively map the areas where you have vegetation. Otherwise, some of these points will fall out of place when you put it on your map, and you will discover that you are actually mapping the wrong location or out of point area in your sampling. So next time when you take your GPS to the site, you have to ensure that it is accurately mapped, that you ensure that it does not go beyond 2 meters. Otherwise, these points will not effectively give you the right information that you are aiming at.
Yes, sir, but I should add that the QGIS software has all these features taken into effect. So the meter approximation you were saying is already
the QGIS that you are talking about, except you are not doing real-time mapping, except you are only using information that were already available online. If you are doing real-time mapping, you have to get the coordinate point so that you compare it with real-time situation on the field.
Okay, noted.
And that's what is supposed to be done to be able to validate whatever information you have on the floristic composition of that area.
Noted, sir.
All right.
Thank you.
Yeah, I have question. If there is an opportunity.
Dr. Yodeji, whom is your question going to?
To the current presenter, the presenter that just presented.
Okay.
Yes.
Quickly.
Yes. Yeah, it's just a kind of an addition to the last person that asked question. I want to ask, does that mean his data is a time series data? Was there any effort made to compare the data he has, if it's a time series, to the practicable data currently, maybe in 2025, for him to arrive at this conclusion? I want to be sure whether it's a time series data or it's a practicable data.
Over to you, the presenter.
Well, sir, these assessments were carried out between 1991 and 2023. And I was also on site to collect the GPS coordinates for the assessment.
Okay. Your essence of being at the site is to collect the coordinate only, not the reflection, because you are doing this study in relation to the plant and land degradation, right?
Yes, sir.
Okay, that is why, because I really want to see your result will be very robust if you have a practicable data to incorporate with the time series data you have studied so far. That's an addition anyway, yes.
Okay, sir. Thank you, sir.
All right, you're welcome.
Thank you very much. In the absence of any other question, I want to thank Dr. Mustafa for the presentation. And I will advise you to harness the advice by the last two speakers to make your work very good, reliable, and accurate. Thank you very much. A round of applause to the speaker. Thank you.
All right, let's welcome on board Dr. Christiana Okay for her presentation. Christiana.
Anybody online from Kwasu?
Yes, sir. Good evening, sir.
Ore?
Yes, sir. I'm here, sir.
Yes. You can go ahead.
Okay, sir. Good evening, sirs. I'm the chairman of the panel. My name is Christiana Aurelua Oke from Kwara State University. I don't know, maybe you can see my slide now. I am presenting on the topic comparative study of two Nigerian beans varieties, productivity through different application of nodumas biofertilization. For the introduction, leguminous plant has been a very good plant that because of the presence of nutrients and other benefits of the plant, and because it's a good plant that helps in fixing nitrogen into the soil. Among these beans variety or cowpea, we have Ife brown and Omo brown. And these two varieties are being gotten from IITA. The Ife brown, I'm coming please. So the study is, sorry please, I'm coming. Oh, I'm sorry, please let me just go back a little. Ooh. Sorry. Okay, so we used one major plant or a fertilizer, which is a nodumat. This nodumat is being produced by IITA, and it has been shown that the nodumat has been used in the promotion and the growth of soya beans. So we tried the nodumat on cowpea to see the effect on the growth and the development, also the yield of the plant. So this study investigates the growth and development response of the Ife brown and Ife Omo to low and high concentration of the nodumat along with the standard. So for the materials and method, the research work was carried out at the screen out of the University of Kwara State University, Malete. The soil collected were collected on an undisturbed land from the depth of 0 to 30 centimeters, dried and filtered to remove large particles. 10 grams of the soil was then filled into polyethylene bags, and the planting was done. Then for the treatment, the planting material, as I've said, the beans variety and the nodumas were purchased at the International Institute of Tropical Agriculture, Oyo State. Then the treatment. Now, the treatment, there is a standard treatment for nodumat in application to seed. But what we actually used here was that we used for low, we tried to modify the producer's measurements. So for the low concentration, we use 0.5 nodumat and the 0.2 gram of Arabic gum for 100 gram of the seed, which was now dissolved in 2 ml of warm water. Then the standard is actually 1 gram of Nodumax 0.2 Arabic gum to 100 gram of seed, while we have a higher concentration for 5 gram, and in that respect also the control was free from Nodumax strain. Then so for the planting was done 10 minutes after the application of the Nodumax. Then 3 seeds were planted in 5 replicates for each of the treatments. The growth was observed for 3 months, and the parameters that were taken, we have the growth parameter, we have the yield, and these plant parameters and details were taken from 5 random selected plants from each of the plots. Now, for the results, one of the things we observed is that the plant that was having one of the Ife brown has a better view. It has a better growth pattern at 5 gram of the Nodumax application than the others. If you can see in the next table that I'm going to present, here in table 1, we can see that the Ife brown variety at high concentration has the stem girth of 15.10, and also the number of nodules were more than the other varieties and other concentration. And the more the Nodumax, the nodules, the more the absorption of nitrogen or fixing of the nitrogen into the soil, and this also helps in better growth than the other plants.
1 more minute. Go to your conclusion.
Oh, okay, sir. So, conclusion, Nodumax inoculation significantly improved the growth performance of both the Ife brown and Omo across the assessed morphological and physiological parameters, which are the leaf area, the leaf number, stem girth, nodulation, and biomass accumulation. These improvements were constantly superior to those observed on the uninoculated control, and even for the standard IITA standard treatment. So the application of Nodumax at a high concentration, which is 5 grams, yielded the most pronounced benefits, particularly in terms of enhanced nodulation, dry biomass, and fresh biomass, underscoring the critical importance of inoculum dosage in maximizing biological nitrogen fixation. Among the 2 cultivars that were used, Ife Omo demonstrated high degree of responsiveness and adaptability to rhizobia inoculation, indicating its greater sustainability for integral into biofertilizer supported legume production system.
Thank you very much, Dr. Okay. A round of applause to Dr. Okay. Thank you very much. Any question from the audience or comment? She has used Nodumax, a strain of Rhizobium bacterium, for maybe nodule formation.
Formation, yes, sir.
I have a question.
It's about fertilizer. Any comment or any contribution?
Okay.
Yes. Can I ask a question, please?
Yes, you can.
You can.
This Rhizobium is usually associated with leguminous plants, and we are seeing how it's able to fix atmospheric nitrogen symbiotically.
Yes, sir.
Are you applying it in non-leguminous plants to see the impact of the bacteria in terms of plant yield and increase in vegetative part of plant, which is very important in meeting the agricultural demands for farm animals?
Hello, sir.
Yes, can you hear me?
Okay, yes.
I said, have you thought about the impacts of applying rhizobium in assessing its impacts on non-leguminous plants? Because we know what we know is that
Okay.
it has leguminous plants, especially. Yes.
I have not tried it on any non-leguminous plants, and according to research that I've carried out or through literature review, this particular nodules has been established in soya beans, but it has not been reported in cowpea. So that was why we tested it on cowpea. But as you have said, we will try and do that on non-leguminous plants, sir.
Thank you very much.
Yes, sir. You're welcome, sir.
Thank you, sir. Any other question?
Yes, I want to present.
There is a question, sir.
Yes, I have a question.
Go ahead.
Okay, thank you. Thank you very much for your presentation.
Yes,
Nodule marks is an inoculum developed by maybe IIT or something for soybean. Yes,
ma'am.
Perhaps I missed it. What was the performance of the uninoculated crops in your treatment?
Okay, for the uninoculated treatment, the growth was not up to the one of the standard. You know, I said that we had 3 treatments. There's a standard which is being used by the IITA. Then that is the producer's manual. But what we did was that we modified the treatment. We used 5 grams of the nodules. The normal standard is 1 gram, but we use 5 grams, and we also use 0.5. We use 0.5 for the low concentration and 5 for the high concentration. And throughout the whole experiment, we observed that the one with 5 grams of nodules did better for both varieties than the IITA standard and the low concentration, and also for the control experiment.
Okay. Yes, that has given me clarity. Where I was driving at is 2 things. Cowpea would usually nodulate with a group of rhizobia called the miscellaneous rhizobia. Cowpea is not selective like soybeans.
Yeah.
Okay, so the increase in the concentration over the standard also means that there was a lot of inoculum in the treatment trials, which is okay. However, are we looking at how to develop indigenous rhizobia so that because these are exotic species, okay, so that we can also, because this cowpea, for instance, is a native African crop.
Yeah.
So perhaps in our treatment, maybe we also isolate rhizobia and then test.
Okay.
Yes. Okay. Isolate rhizobia from our cropping systems.
Okay.
Right? Depending on the cropping history of the soil or of the area. Depending on the cropping history. Then we can also compare with nodule marks.
Okay.
And see what happens.
No problem.
Yes.
We'll work on that.
All right.
Thank you so much for your
contribution. Thank you very much.
Please,
yes,
please, the last question for the presenter. Can I have that? Okay. In next presentation, Christiana, okay.
Yes,
I have a concern with your title and the work generally. Looking at what you consider between April to June.
Yes, sir. 3 months.
3 months.
Yes,
for you to make this general assertion about the nodulation of legumes, you will understand with me that seasonal changes have a significant impact on nodulation.
Okay, sir.
And if you consider that, it will be better for specificity's sake and for you to be very, very specific, that your title should have rather indicate the season under the consideration.
If you look at your work, you have presented on between the month of April observation, May observation, and June observation, which only covers just the wet season. In the dry season, the scenario may play out differently.
Do they crop in the dry season?
Thank you very much. Thank you very much. Well, there should be seasonal variation anyway. During rainfall, we will have more nitrogen being made available through physicochemical means of nitrogen fixation during rain and lightning.
Okay, sir.
So, thank you very much. Although you didn't give any background to the nitrogen content of the soil. So that might have accounted.
Yes, we actually did the nitrogen, the mineral nutrients of the soil.
It's okay.
Sorry,
Thank you very much for the beautiful presentation. A round of applause for the presenter, Dr. Oke. Thank you. The next person.
All right. The following person should get ready. Dr. Adedoyin
Please,
Yusuf, Dr. Eunice Oyedede Babalola, and Dr. Lema Aisha Mohammed. So the next presenter we'll be having is Okwadokun Wasiu.
Wasiu, Dr. Wasiu Okwadokun.
I have a question.
Dr. Wasiu Okwadokun from Alkima University, Ilorin.
Good evening, the chairman. All protocol duly observed.
Please, you should be calling this so that we know which number is presenting. Which number is
All protocol duly observed. My name is Okpadokun Wasiu Olanrewaju. I'm presenting on effectiveness of S-metolachlor and nicosulfuron on weed control efficiency and nutritional quality of disease. Maize is a key staple crop in sub-Saharan Africa, but weed infestation can reduce yield by up to 80% if unmanaged. Traditional hard weeding has been used, but is constrained by labor shortage. However, farmers use herbicides such as S-metolachlor and nicosulfuron for effective weed control. These two herbicides, they are used by farmers in control of weed. The studies evaluate these herbicides for their effect on weed suppression and maize grain nutritional quality. Statement of the problem. Control is considered both effective and commonly viable for maize production, and it reduces weed competition and enhances yield. However, the use of S-metolachlor and nicosulfuron can pose risk, including food contamination, soil degradation, and disruption of nutrients. Despite all this, there are limited field studies that have assessed their combined effect on weed control and grain production. Maize justification of the study. Maize is a vital food security in sub-Saharan Africa, but herbicides used, though effective for weed control, can alter its nutritional content and pose serious health and environmental risk. However, it is necessary to investigate the effect of these two herbicides on weed suppression, maize grain quality, crucial for ensuring safe and sustainable agriculture. The aim of the study is to know the performance of S-metolachlor and nicosulfuron across two cropping seasons, with emphasis on their impact on weed suppression and proximate and mineral composition of the grains. So these objectives are to that of weed control efficiency and proximate and mineral content of the harvested maize. Materials and methods. These herbicides were sourced from a suitable maize cultivation in tropics in Kwara states and Maxwell Agrochemicals, and the maize grain were obtained from IAR in Ahmadu Bello University, Zaria, and the variety of maize that we used was Swam 1. The experimental side was conducted at Rewumi community in Ilorin, and the field experiments preparation and the experimental design, it was weed and laid in complete randomized block design, and we have different treatment details. We have a control that we don't use any herbicide, and we have two herbicides of two different levels. The first one is to use S-metolachlor at the recommended rate, and we use double the recommended rate for the S-metolachlor. We use nicosulfuron recommended rate of 1.5 liter, and we double it by 2, and we have the zero weight control. However, soil physiochemical parameters were taken using the following parameters, and we take some weed parameter too by checking the weed vigor, relative abundance, weed density, biomass, and weed control efficiency. So mineral elements and proximate composition were also determined, and it was subjected to statistical analysis. Now, these are the weed present on the field, and from these results, most of the weed present here, they are broad leaves. Now, from the weed density, we notice from the results that plots treated with 3 liter per hectare of nicosulfuron significantly reduced the weed from the table, followed by plot treated with 1.5 liter per nicosulfuron when compared to others. Also, we check the dry weight. Plot treated with 3 liter per hectare of nicosulfuron drastically reduced the dry weight. Likewise, the weed control efficiency, it was drastically reduced in plot treated with 3 liter per hectares of nicosulfuron, which have the highest weed control efficiency. Then from the proximate composition, we notice in the 2 cropping seasons that plot treated with nicosulfuron produce the highest crude ash, crude fiber, crude lipid, and carbohydrate content. Likewise, the analysis of macro elements present, we notice that plot treated with 3 liter per hectare produce the highest number of calcium, potassium, magnesium, and phosphorus. Now, from the discussion, we notice that we have different species of broadleaves that are present, and the dominance of broadleaves can be attributed maybe to favorable climatic conditions such as adequate moisture, moderate temperature, and high humidity, which support their growth. The decrease in the dry weights was evident in all herbicide-treated plots and can be due to the effective and efficient of the herbicide. However, the control efficiency of the
Your
weed
time is off, please. Conclude in a sentence. Conclude in one sentence.
Thank you, sir. The application of nicosulfuron at 1.5 and 3 liters consistently resulted in effective weed suppression and notable improvements in both proximate and mineral composition of maize grain across the 2 cropping seasons. This application rate provides superior to the other herbicide, which is metolachlor, making them more suitable for enhancing weed control and nutritional quality in maize cultivation. Therefore, the use of nicosulfuron at this rate can improve yield of maize grain. Thank you, sir.
Thank you very much. A round of applause to Dr. Wasiu Olarewaju Okpadokun. Thank you very much. Any question for the presenter? Thank you.
But normally, that's not where we're supposed to.
Thank you, Dr. Waziri, for your presentation.
Your concentration should
be. You know, as a botanist,
if you do not show me,
we'll be speaking against the use of synthetic chemicals in the environment because we know the effect in the environment. So my question is this. Could there be any,
Or the other one is what?
could I say plant extracts or whatever that could be used in place of synthetic chemicals in the environment? Thank you.
Thank you very much, Ma. We have many plants that can be used in place of it. A typical example is Titonia diversifolia. Titonia diversifolia have been reported that it has a lot of allelopathic effects. Whenever you see that particular plant, you notice that you see plants growing. Very rare you see plants competing with it whenever there is plant around it. And this particular plant, it has been recorded that they have allelopathic effects, and research has been going on by taking getting this allelopathic that is present in this particular plant. Instead of using all these synthetic herbicides, I think that particular plant is another alternative because of its allelopathic potentials.
Thank you very much, Dr. Wasiu Okpadokun. A round of applause for. Wait for a question from Dr. Yedeji.
Dr. Okpadokun, thank you very much for your presentation.
Yes, sir.
This nicosopron you talked about is a synthetic chemical.
Yes, sir.
I'm just a little bit worried that you may need to check for residual effects of this chemical in the maize grain as not to also contaminate it, despite that you might have gotten improvement in the proximate and nutritional composition of the maize that you use this chemical to actually suppress the weed in the plots. So it will be very, very important to check the safety in terms of the chemical residual effect on the maize that you obtain from the plots.
Thank you very much.
Okay.
As a rider to that, not only in the seed of your crop, but also in the soil to prevent injury to any crop you may want to plant on that same plot of land at a later time. Thank you very much for the presentation.
Thank you, sir.
A round of applause once again for the presenter. Thank you.
Thank you very much, sir.
We have the next presenter, Adedoyin Yusuf.
I have a question though.
Adedoyin Yusuf, it's your time to present.
The next presenter, Dr. Adedoyin Yusuf. Adedoyin Yusuf.
The absence of Adedoyin Yusuf, the night presenter.
I'm here now, sir. It's my network.
Are you available to present to us?
Yes, sir. Yes.
Okay, go on.
Yes, sir. I'm available. My network, sir. I want to share my slide now, sir.
While you share your slide, you can start because your time has started counting. You have just 7 minutes.
Okay. Good afternoon. Good afternoon, everybody. I'm Adedoyin Yusuf from Biological Science Department, Bamidele Orimilwa University of Education, Science and Technology, Ikere-Ekiti. I'll be presenting on effect of diesel-simulated polluted soil on the growth of Phaseolus vulgaris as a test crop. Here are the outlines of my study. Introduction. Oil spillage is one of the contagious issues in Nigeria, especially in the contamination of soil. Soil pollution by petroleum products, especially diesel, are presently a menace in Nigeria. The exposure of plants to diesel and heavy metal poisoning has been reported to produce reactive oxygen species and other free radicals which induce oxidative stress and causes lipid preoxidation. Even at its early stage, it causes reduction in cell proliferation and growth. Phaseolus vulgaris is the most important food legume for direct consumption in the world. Hence, the need to determine the effect of diesel pollution on Phaseolus vulgaris germination. Here are the objectives of my study: to investigate the performance of these bean species on a diesel-contaminated soil, to determine the viability of the seed grown on diesel-contaminated soil, also to determine the tolerance of the concentration of diesel-contaminated soil on Fusarius vulgaris. Materials and method. The experiment was conducted in his greenhouse at Bamidele Olumilua University of Education in Ekiti. The experiment was designed in accordance with the modified method described by Adewale and Haboye 2015. Box 0 to 15 centimeters were collected in an area in the university. He had dried for 3 days, sipped under 2 mm, wired sieve, and analyzed using the standard method. 12 polythene pots with drain holes at the bottom, each containing 5 grams of soil sample, were randomly placed in the greenhouse using a factorial combination of 5 treatment level of 0 degree to 40 degrees C of diesel, and designed in pots 1 to 5 respectively. 3 seeds of beans per plot were planted. The bean stand were regularly watered throughout the growing period. Then at 2 weeks of the planting period, the beans were thinned to 2 stands per plot, and the thinned stand were retained inside the plot so as to maintain the nutrient that might have been lost during the process where the stand of the bean were removed. So fortnightly, the growth parameters of the beans were measured. The plant height, the leaf counts were measured till 28 days when the experiment was terminated. After the termination, the roots and shoot length and weight of plants were carried out using thread roots and weighing balance respectively. The roots were also separated from the plant, and then measurement was taken. After the weight of the fresh plant was carried out, it was then oven dried for 48 hours at 80 degrees Celsius in order to carry out the dry weight sampling. The table 1 shows the effect of different concentration of diesel contaminated soil on plant height. From this table, it was observed that in plot 1, across the 4 weeks of the planting period, in plot 1, the result shows higher value for the plant height throughout the 4 weeks planting period for plot 1, while plot 1 containing a 0 gram of diesel, which is the control medium, while for plot 5, a reduction in the height of the bean species were observed across the 4 weeks. Also, similar results was observed on the effect of the diesel contaminated on the average fresh weight of the plant species. Similar results was observed like that of the plant height.
2 more minutes. Go to your conclusion.
All right, sir. The conclusion of my study, from my findings, the experiment revealed that there are phytosociological dysfunction in beans plants as a result of the addition of diesel to the plant species. It was observed from the experiment the plant species that have been treated with 0 grams of diesel were observed to have a higher plant height, dry weight, and for the leaf count, there are higher values of this recorded compared to those due to the treatment in which a higher concentration of diesel were being added to the planting. While the recommendation for the study, further research into cost-effective and suitable remediation strategy for diesel-contaminated soil. There is urgency for government and environmental agencies to enforce stricter policy on petroleum handling. Thank you for listening.
Thank you very much. Thank you very much, Dr. Adedoyin Yusuf. A round of applause for the presenter, please. Any question or comment? Thank you, sir.
Please, you never told us where you collected or you bought the Phaseolus vulgaris, and possibly information that has to do with the identification.
Okay, sir. The Phaseolus vulgaris was bought at a market in Ikere Ekiti here, and it was identified in the herbarium of the Bamidele Olumide University of Education, Science and Technology, sir.
The market, is it from one of the bean sellers there, or it's a seed shop that any time anybody can go there to procure the seeds?
Like a seed store, sir, where like a seed bank, where like a local seed bank where plants can be collected to carry out experiments, sir.
So you have been advised to be much more scientific. A local seed bank. Thank you very much for the presentation. Yes, sir. In a minute. Steve, hold on.
Thank you, sir.
Okay, sir.
Thanks for the presentation. Please, I'm just worried that this Phaseolus vulgaris, we tested the ability to grow on contaminated soil, diesel contaminated soil.
Yes, sir.
So I'm now wondering, is it
Yes,
that the place where they grow this plant, diesel are usually there, or I just don't know. Why testing the ability of this plant on contaminated soil?
Okay, sir. Because Phaseolus vulgaris is a widely consumed. It was a widely consumed legume plant, and it also contributes to food security and soil fertility. So, hence the curiosity to determine the effect of a certain pollutant. So, I try to choose a diesel to carry out the research using diesel as a pollutant to see the effect of diesel on Phaseolus vulgaris, because it's a widely consumed leguminous plant. So I want to see the effects of the consumption of the effects of contamination of diesel on the plant, sir.
Well, thank you very much. But since you said
thank you, sir,
this crop is arable crop to the people there, you wouldn't have used this for bioremediation of polluted area, but it's good. It's still scientific to carry out to know whether it will bioremediate or exclude the pollutant, and also whether it will be able to extract it, or it will translocate. So maybe you use other plants rather than this. That you the experiment is scientific, is good. You can go ahead. Thank you very much.
Okay, sir.
A round of applause.
Thank you, sir. Thank you, sir, for your advice, sir.
The next presenter we have on the list is Dr. Eunice Babalola.
Dr. Eunice Babalola, are you with us?
Yes, I am. I am with you. I want to share my screen.
We are still waiting for you. Should we go to the next person?
No, I'm sharing my screen. Hey, is my screen showing?
Yes.
Okay. Good evening, distinguished guests, my fellow colleagues. My name is Eunice Ayodele Babalola, and I'm here to present a paper on the effect of ethanolic and aqueous extracts of Cucuma longa on Tetradesmus obliquus growth and fat composition. First of all, let me introduce the plants, Tetradesmus obliquus. First of all, it's a green algae under the phylum Chlorophyta. It's on the family Cnidesmaceae, and it is a photosynthetic algae and an aquatic algae usually found in fresh and brackish water. The morphology of this plant is that the cells are usually elongated. There's a photomicrograph showing of my Terresmus obliquus cells. You can see that the cells are usually elongated without spine. And in this species, this genus, Cnidesmus and Tetradesmus, their morphology is very variable depending on the liquid, on the culture where they are found in and their environment. Then it's also a colonial microalgae. It has presence of colonies, which are usually called cyanobia, but it also has presence of unicells in when it's growing in a liquid form, in a liquid culture. Then this plant, why it's of very big interest to us, is that it yields a large amount of lipids. It's very high in lipid content and bio-oil content. Then another plant we are working with is the Cucuma longa. This is called turmeric. And in this work, for the sake of this work, I looked at it as yellow turmeric. And this plant is a member of the Zingiberaceae family. It's a herbaceous perennial plant. And in this work, we used the rhizomes of the plant. We worked with the rhizomes of the plant. And the rhizome of the plant is usually color from yellow to orange. That's why we call it yellow turmeric. And it's very rich in nutrients, vitamin, and mineral. And based on literature review we did on this plant, we realized that the plant has, when other plants are treated with the extracts of this plant, it helps to improve those plants' growth parameters and also improves the nutritional value content, like the protein, the fat of the plants that are grown in it. So why this work? Well, because nowadays there's a need. There's a need for an alternative to basically a lot of products to use that are affecting the environment. There's a need for green, non-toxic alternative bioproducts. And even though algae is a perfect candidate, it has always had problem entering into the market because it has a very high cost of production. So it's always been deemed non-feasible. And some of the challenges that we face with is nutrient sourcing, productivity, and utilization of biomass, and harvesting. So to increase productivity, researchers have tried to use vitamins, growth hormone, and different plant extracts. And generally, vitamins and growth hormones sometimes are not readily available, and they are also very expensive. So the point of this work was to try and find if you can use cheaper, readily available plants like turmeric as their extracts to see if they can help support the growth and, most importantly, support bioaccumulation of certain bioproducts in the plant. So the aim of this study was to investigate the effects of this extract on Cucuma longa on the biomass and fat composition of Terresmus obliquus. So the water sample we used to get this microalgae was collected from the fish pond at the Department of Zoology and Environmental Biology at the Zoological Garden, University of Nigeria, Nsukka. And when we got the water sample, we viewed it under a microscope, and we were able to identify the microalgae present in it. And Cnidesmus obliquus, we identified it morphologically as Cnidesmus obliquus. Then we grew it in BBM, bold basal media, and then we went on to do to get an azenic culture by streaking, plating, and subculturing of the samples. Then, using DNA analysis, we're able to morphologically identify it as Tetradesmus, as molecularly identify it as Tetradesmus obliquus. Then the Cucuma longa was obtained from Massacre Market in Nasarawa State, and the identity was confirmed at the Herbarium Department of Plant Science and Biotechnology, UNN.
Thank you very much, Dr. Babalola. Tell us your conclusion.
Yes, sir.
Conclusion, please.
Okay, sir, I'm coming. Okay, at the end of the experiment, because we did the experiment for 14 days and we checked growth every 2 days for 7 times of checking the growth. At the end of the experiment, we observed that the extracts supported, both aqueous and enterologic, supported the growth of the T. obliquus. But on the case of the ethanolic extract, the yellow turmeric ethanolic extract, comparing to the BBM, there was not any significant difference in the population at the end of the 14th day. But the 2 ml of the yellow turmeric extract supported the growth the best. Then on the case of the yellow turmeric aqueous extract, there was a big significant difference. The BBM grew supported the population growth of the T. obliquus better than the aqueous extract. So it was significantly different than the aqueous, the population of the T. obliquus grown in the aqueous extract. Then, on the basis of the fat biocomposition, we observed that the algae biomass that were treated in the yellow turmeric ethanolic extract had a higher fat composition compared to the control, which is the basic T. obliquus fat that we checked. And we observed that the 8 ml had the highest percentage of fat at 12.5% of fat bioaccumulation. So from our results, we can say that these extracts are actually good promoters of algae and T. obliquus biomass growth. And we observed that it supported the fat bioaccumulation of in the 8 ml of the extract. This may be due to increase in concentration of the nutrients. It also may be due to light intensity, because light intensity in microalgae can affect fat bioaccumulation. But and the conclusion is that the extract of this sea longer showed appreciable promoter activity, and the highest fat percentage was found in the yellow turmeric ethanolic extract. This study supports the idea that plants and their extracts could potentially be used to fortify microalgae media in cultivation and can be used as added strategy to support production of desired biocompounds. But further studies should be done on the extract concentration that supported or increased production of bioactive compound in T. obliquus higher than the control. This could play an important role in boosting the industrial and commercial application of microalgae for food and nutritional supplements and the source of biofuel. Thank you for listening.
Thank you very much for your presentation, Doctor. A round of applause for Dr. Babalola. And let's stop. The one you have given to us is informative enough. Thank you very much. Any question?
Please, you have skipped my number.
Go ahead. Any question? If there is no question, we are very, very grateful for your beautiful presentation. A round of applause once again to
Eunice. Thank you, sir.
So we go on to the next presenter, Dr. Lema Aisha Mohammed. Dr. Lema Aisha.
Are you online with us? Dr. Lema Aisha Mohammed.
Online.
Are you with us?
Dr. Deborah, please get ready.
This is Dr. Kanaye, please. Ready, please.
Good evening to you all. I'm Lema Aisha Muhammad with a presentation on evaluation of morphological trait and lycopene content in tomatoes varieties. Tomatoes belong to Solanaceae family, most important vegetable crops after potato, a rich source of vitamins, minerals, and antioxidants, particularly lycopene. Morphological trait, describing plant species based on their observable trait. Lycopene, natural occurring carotenoid, responsible for the red color in tomatoes, watermelon, and pink grapes fruit. Potent antioxidant with the ability to reduce the risk of cancer, cardiovascular diseases, neurological diseases, diabetes, and so on. The aim of this study is to evaluate morphological trait and lycopene content in tomatoes variety. The objectives are to determine the morphological characters of both fruit and shoot of selected varieties of tomato. Then determination of lycopene content in fruit of selected varieties of tomatoes. The content of lycopene is influenced by the following varieties. Naturally, some varieties have the ability to accumulate more lycopene than the others due to their genetic makeup. Growing conditions such as the soil, temperature, water, and so on can affect the content of the lycopene. Ripeness. Typically as tomato ripen, it accumulated more lycopene. Then processing. Different processing methods can affect the content of the lycopene in tomato. Then biosynthesis of lycopene. The biosynthesis of lycopene begin with the general diphosphate, then biotin synthase, biotin dicitrate, zeta-carotenoid, zeta dicitrate, neurophosphate, carotene isomerase, and lycopene. Methodology. The study was carried out in the Botanical Garden of Usman Danfodio University, Sokoto. Sample collection. The samples were collected. The sample of improved local and hybridized variety were collected from satisfied vendors in Sokoto local market. Soil preparation and planting conditions. The seed were first sown into nursery bed. After 4 weeks of planting, the seed were transplanted into plastic container containing a mixture of rice husk, soil, chicken droppings, and NPK fertilizer in a right proportion. Experimental design. The experiment was carried out in complete rhizomal design in 10 replicates. Each consists of one plant. Determination of morphological character. The morphological character will be determined at the vegetative level and at the maturity stage. At the vegetative level, in 3 replicates of the tomato will be harvested and root will be washed. Then the plant height, root length, and the leaf area will be measured using ruler. Then the leaf area will be determined using graphical method. Number of the leaves will be counted. Then at the maturity stage, the number of the flower will be counted. Then the fruit shape and size and color will be determined using
Thank you very much.
standard
Thank you very much, madam. Let's go to the conclusion of the work.
Okay. And the conclusion reveals that varietal selection is crucial for changing morphological content and desirable trait in tomatoes. As we can see from I have not presented, from our result at the vegetative stage, most of the morphological characters show non-significant differences, but Chibli and OXs exhibit large fruit size, and all the other varieties show red color with the exception of Bahaushi and Gada. They are the local varieties which tend to be bright in color. And it is a recommendation. You prioritize varietal selection in breeding program and utilize high lycopene varieties like UTC for nutrient-enriched product. Thank you, sir.
Thank you very much for the beautiful presentation that encouraged us to always make good selection if you want good harvest for our food security move. A round of applause to the presenter, please. Any question for her? Yes, madam.
All right, good presentation. What I would like to ask, was there any correlation between the ripeness method of processing and the morphology with the lycopene content? I think that is what I expected to see in your conclusion.
I didn't get you.
Was there any correlation between the ripeness, method of processing, and morphology of your tomato fruit with the lycopene content? That is what we expect to see in your conclusion.
What you expected to see in the conclusion, we just selected fully ripe varieties of tomatoes and we performed the determination of the lycopene content in fresh and dry form of the tomato. And the dry form of the tomato is having the highest content of lycopene than the fresh content of tomatoes. And UTC has high content of lycopene in both fresh and dry content of tomatoes.
Yes, that is good. But was there any correlation between the degree of ripeness? Because you mentioned that the factors affecting lycopene content could be the variety, the ripeness, the soil and environmental conditions. So did you derive any correlation between all
No,
these?
we did. We just take the fully ripe tomatoes, though we didn't take those that are not fully ripe.
Okay, thank you very much. Maybe in any other subsequent experiment, you can try to see how the degree of ripeness and the lycopene content.
Typically,
just an advice.
Tomato wrapping.
It's an advice.
Okay, thank you,
in
sir.
subsequent ones, maybe if you have other students, check this place, look at this place, and thank you very much for giving us this beautiful presentation. I think the mommies around us will love your presentation, and a good take home that selection is crucial to put tomato production. Thank you very much. A round of applause for her once again.
Thank you, sir.
I think we have, sir, we only have one more, I don't know. Deborah, are you online?
Yes, sir, I am.
One.
All right. Good evening, distinguished colleagues, great Bosonite. I am Dawende Ezekiel. Let me start on the already established protocol. I am a postgraduate student from the Department of Plant Science and Biotechnology, University of Jos. So the topic for my presentation is proximate composition of Solanum tuberosum varieties infected with Fusarium species treated with Syzygium aromaticum based biofungicidal formulation. So, as a way of introduction, Solanum tuberosum is commonly known as Irish potato and is ranked for as among the most important staple crops globally, with an annual production exceeding 375 million metric tons. Of course, potato is vital, it's a vital crop, especially in temperate regions such as Nigeria, due to their caloric expanding role in enhancing rural livelihood and nutritional security. Fusarium-induced vascular wilt are, of course, in the field, and dry rot in stored potato. These fungal pathogens diminishes marketable yield and significantly alter the nutritional composition of tubers. Yes, sir, I am presenting already. Hello? Sir, are you getting me?
Yes, go ahead.
It's okay. All right, thank you. So, can you see my screen?
Go ahead. You have less than
it's okay.
4 minutes to present to us.
It's all right, sir. So the aim of the study was to determine the proximate composition of S. tuberosum varieties that were infected with Fusarium species, and then which was treated with Syzygium aromaticum based biofungicidal formulation. Of course, the specific objectives are there on the slides. The methodology adopted was to isolate the fusarium from 3 well-known potato-producing areas. So the Syzygium aromaticum, which is commonly called clove, was obtained from foreign gather markets and then extracted by maceration. The experiment was conducted under greenhouse setup, and the potatoes were obtained from National Root Crop Research Institute. The experimental design was a 4x4x3, giving us a 48 treatment combination. The parameters determined were moisture content, crude fiber content, protein, fat, ash content, nitrate-free energy and metabolic and energy, calcium and phosphorus. So the data generated were subjected to analysis using complete randomized design, and they were analyzed using Microsoft Excel 2024 version. T-test was used to determine compare the proximate values across treatment and location. And then for separation of means, Turkey's post hoc was used. So the results obtained showed that the mango and does not isolate had significant nutrient improvement after treatment, whereas that of Berkin-Ladi had no significant difference with respect to the parameters measured. Nitrogen-free extract was decreased, indicating an altered carbohydrate metabolism. So these are the results, the proximate analysis. So for the discussion, like I said earlier, mango and does not isolate has significant proximate and nutritional difference for the agroecological zones from where the isolates were obtained. Of course, there was a deviation as far as Berkin-Ladi isolate was concerned. Now,
thank you very
results
much,
actually
madam. Thank you very much. Your
okay.
materials and method,
yes.
Your discussion has given a very good logical conclusion. Now give your conclusion in a sentence.
So, all right, sir. In conclusion, the study contributes significantly to the growing body of knowledge supporting eco-friendly, low toxicity alternatives to chemical fungicides, integration of Syzygium aromaticum-based treatment into potato post-harvest protocol holds a promise for improving food safety, enhancing nutritional outcome, and advancing sustainable agricultural practices, particularly in sub-Saharan Africa. Thank you. And I also would recommend that further studies can be conducted in determining the mechanism of action of Syzygium aromaticum. Thank you.
Thank you very much for the brilliant presentation and the assurance and confidence put into the presentation. A round of applause for the madam. Any question for her? Thank you, sir.
Just a comment. It seems I heard you say temperate regions such as Nigeria. Then your microorganisms also, maybe...
Tropical, sorry.
Okay.
Tropical, sorry, sir.
Then your maybe microorganisms, which institute that you say you collected them from also? Or is it the potato? You mentioned something about collection or institute or something, whether it's the Irish potato or maybe you use microorganisms. I wouldn't know.
Are you clear with
yes.
the question?
Network is a bit... I actually didn't get you clearly. My network is a bit bad.
Where did you make your collection?
Hello?
And where did you make your collection?
Okay.
And then did you use Irish potato or sweet potato? Irish
potato, Solanum tuberosum.
Okay, where did you collect the samples? Where did you collect the samples?
The potato or the organism? For the organism, which is Fusarium, I isolated them. It was isolated from soils in these 3 locations I mentioned known to have cultivated potato for at least 3 farming seasons.
The location of collection.
Mango, we collected microorganisms from mango soil, Barikin Ladi soil, and just nuts.
Thank you very much. A round of applause for the madam.
Yes, sir.
Thank you very much.
Sir, I have a clarification also.
Okay, sir.
Yes,
SB, 1 minute.
Yes, sir.
1 minute.
Yes, sir. She made mention of the differences between those from mango and just nuts, which is understandable. But what about Barikin Ladi? Because when you look at Barikin Ladi, inside Barikin Ladi in Doroa, so it's very close to mango. Did you experience differences in those samples too?
Yes. As a matter of fact, the organisms were characterized. So based on the findings, the isolate from mango and just not were Solanium, Fusarium solani, while that of Barikin Ladi was Fusarium falciforme.
Yes, that's the clarification I want to see, because Barikin Ladi, where they grow potato in Barikin Ladi,
it's okay.
It's very close to mango, because when you go inside Doroa Soho, it's inside. Even you can even walk from villages in Doroa Soho to mango. So I'm just seeing why differentiation. We understand if there is variation between just not sample and mango sample, fine. But with Barikin Ladi and mango, because they're very close by, so I don't know. Maybe you can explain for them, maybe in your paper or something.
Yes.
Why the variation?
Yes, okay. The essence of the variation is because we understand that these areas where potato are actually planted, of course, there has been overhype of Phytophthora infestans. However, in the course of researching, we stumbled over the fact that most of these potato growing areas have a lot of fusarium in their soils. So it was just sheer inquisitiveness to know whether the organisms are the same as far as these locations were concerned or not. As a matter of fact, the just not was substituted for Bocos, because Bocos was the actual place we wanted to take the sample. However, because of insecurity, that was why Bocos was left out.
Which part of Barikin Ladi did you take the sample?
Close to the university. Close to the university, sir.
Okay. That's Foron district, right?
I actually did find out whether that is Foron district.
Okay.
But I think it is. I'll find out.
Okay, thank you very much.
Okay.
Yes. You're welcome, sir.
Let her go and find out. Thank you very much for the contribution. SB.
Thank you so much, Prof.
Thank you, sir.
Okay. Contribution or question? Okay. So,
okay, thank you very much for this.
Sorry, my network is bad. Am I still expecting more questions?
It's okay. Do you hear me? Can you hear me?
Okay, yes, sir, I am. I can hear you now.
Okay, this identification of the species, at what level did you identify them? Is it morphologically or physiologically?
Molecularly. We did a molecular study of the isolate.
Okay, that's what I want to hear. Okay, thank you very much.
You're welcome, sir.
The time is against.
Yes, the time is against her, but the university she mentioned is in just south. I think you need Jos, right?
Yes, sir. University of Jos is in Jos North, not Jos South.
Jos North. So I asked you where you took the sample in Barikin Ladi. You said close to university. So I've been thinking about
that's
that.
Plasu. Oh no, that's Plasu, sir. Plasu State University.
Okay. That's well, it's close to Mangu too. All right.
All right, please, just a contribution. I think it is also important you put the GPS location of your points of collection. That will actually make it more scientific for presentation.
So thank you very much for the brilliant presentation. A round of applause once again for her. Thank you. With this, we are closing for the day. Thank you very much, sir.
Madam, we are closing for the day. Inshallah, Madam, machine is not emitting so that at least when you're talking, they will.
Thank you, Professor Olorumaye and the rapporteur for a wonderful anchor of the online session for today. And on behalf of the Executive Council of BOSON, I would like to thank everyone for staying up to this present moment, this past 6. And by tomorrow, we'll be starting exactly 9 a.m. There will be physical classes tomorrow, and we'll continue also with the virtual and online session tomorrow also. We want every one of us to be here by 9 a.m. so that we'll start as early as possible and continue with the other programs we have tomorrow. I think tomorrow, some things have been on schedule for tomorrow. We are going to have our AGM tomorrow. And before the AGM, if you are able to finish our presentations, there's likely to be a field excursion. Thank you. Thank you very much. Thank you. I want to Professor Akoma now.